A big hello to all my Chutku people. How are you all? I hope everyone who is watching this video is doing absolutely good. You're not at all worried about biology exam because it is going to be really good, right? It is going to be good. So let's have a positive affirmation and let's begin with top 80 plus questions part 3. And today I am going to complete, or it's not like I am going to complete, it's we are going to complete the last 5 chapters: human health and diseases, microbes and human welfare, biotechnology principles and processes, biotechnology and its application, and organism and population. Done. Then we are done with all the questions. So where are these questions taken from? These questions are taken from the question bank, my dear children. Where do we find the previous year questions, ma'am? Those I have solved in the 2 marker, 3 marker and the 5 marker series. So if you have not watched it, watch it. So what is the pattern that you should follow is, you take the NCERT, read the chapter once, just give yourself 15 to 30 minutes, not beyond that, just for reading the NCERT. Once you're done with reading the NCERT, take the MCQ questions, okay, from the 1 marker. I have divided class, chapter wise. So see the video of 1 marker, then come to 2 to 3 markers, see the chapter, 5 marker, see the thing, and then come over here, question bank. That's how you should be making the concepts really thorough. You might find similar questions here and there, but it's not very common to find. So I have tried to include those questions which you might not find it there, and some question which I feel is very much important I might have repeated as well. Yeah, so are we ready? I hope we are ready. Let's begin. No more talking. I talk a lot, I know that. Yes. So human health and diseases, it carries 11 marks in your examination. Do you know that? So let's take into priority and let's see the question. Name the host and the site where the following occur in the life cycle of malarial parasite. First tell me, what is a malarial parasite? What causes malaria? It is Plasmodium, my dear children. Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae causes what? Malaria. Okay, and the malignant form of malaria is caused by Plasmodium falciparum. If the question comes, you have to write this. Now, you are asked, name the host. Okay, host as in where is the organism, where does this particular parasite live in, and also the site where the following things are happening, that is formation of gametocytes. First thing, what do you mean by gametocytes? Gametocytes are cells or is a stage. Okay, gametocyte is a stage where it can possibly divide and form gametes. It can divide and form gametes. Okay, so in human body, gametocytes are being formed. So I can say the host is human for sure, but where is the site in the human body where these gametocytes are formed? It is RBC. RBC is also known as red blood cell or erythrocyte. Listen to me, gametocytes are formed over here, but gametes are not formed in human body. Gametes are not found in human body. So in RBC, these gametocytes are found. Okay, RBC bursts open and every gametocyte is being released in the blood. Now what happens? I am a mosquito, I come and drink it. I am a mosquito, I come and drink it. Okay, I take the blood inside, it goes to my gut. In my gut, I have the gametocytes as well along with the blood. The gametocytes will now give rise to gametes, and they will also fuse there in the mosquito's gut. So we have fusion of gametes. The host is what? Female Anopheles mosquito. Where is the site? It's the gut of the mosquito where it happens. Now, let me tell you the next story. Okay, so they have fused, they have formed something that is known as ookinete and things like that. There are so many stages, my dear children. At the end, from the gut, they enter into the salivary glands of the mosquito in a stage that is known as sporozoid stage. What stage? Sporozoid stage. Okay, now it's where? In the salivary glands of the mosquito. Now suppose you are the human being, I came to bite you. Okay, when I came to bite you, what did happen? I basically am going to release my saliva because my saliva has anti-clotting things so that it does not allow the blood to clot and I can drink your blood peacefully. But what happens in my saliva? There is sporozoid also. These sporozoids will enter your body, and when they enter your body, they will go to the liver cells first. They will go to the liver cells first. They are going to have different stages of it. Okay, it's not the sporozoid which will be there in the here and all. There will be different stages. Okay, now in the liver, they will again divide the liver cells, that is hepatocytes will burst open, and now again they are going to enter the next set of cell, that is RBC. In RBC, they are going to form into the next stage, that is gametocyte stage. Okay, now in gametocyte stage, as I told you, it will be again the RBC will rupture, gametocytes are released in the blood, but these gametocytes are not going to form gametes in our body. It will happen in the midgut of, not in the midgut, basically the gut of the female Anopheles mosquito. I hope it's clear. One more information I'll give you regarding malaria only. When RBC bursts opens, my dear children, there is a very toxic thing that is getting released, that is known as hemozoin. So what happens is that the Plasmodium, it feeds on hemoglobin. It feeds on hemoglobin, which is there in the RBC. Now hemoglobin will be broken down to heme and globin, and the plasmodium will be like, I don't like the heme, it is kind of toxic for me, I have to convert into something else. So for itself it converts into hemozoin, and it's there in the RBC, so that it's not toxic for it. But when the RBC bursts open, the hemozoin is released, that thing is toxic for us, which is why we get fevers and chills. Okay, so I hope you got the answer. Formation of gametocyte, host is human, site is red blood cells, and fusion of gametes happens, host is female Anopheles mosquito, and the site is gut of the mosquito. I think you get lots of questions cleared with the help of this one question, which is of 2 marks. I have explained a lot. Now, yes, mention the types of innate barriers of innate immunity. What do you mean by innate immunity? Immunity that we have from childhood, from the time we are born, that is known as innate immunity. It is not specific type of immunity. For example, in the case of acquired immunity, we have 2 types of immunity, innate and acquired. In acquired immunity, we acquire it throughout our lifetime. For example, I catch a cold and I develop resistance to something. Okay, I catch a disease, I develop a resistance to something. Next time, the same disease won't be happening to me. You know, that is acquired immunity. Innate immunity as in, for example, there is no specific goals. I don't know what I have to kill to. For example, skin is a part of innate immunity. If a bacteria tries to get in, if a virus tries to get in, if a fungi tries to get in, everything it will stop. It's not like specific. It will be common for all. You get it? So over here, the types of innate barriers in innate immunity is physical barrier first. So it prevents the entry of pathogens. Example, skin, the mucous membranes of respiratory, gastrointestinal and the urogenital tract, one thing. Second is physiological barriers. For example, it maintains, what do they do? It maintains unfavorable condition for pathogen survival. For example, my tears, okay. My tears, crocodile tears, yeah, acidic pH of the stomach, and tears because it contains lysozyme, and lysozyme basically breaks open the cell wall of bacteria and all. That's why they are not able to survive. Body temperature, all of them are physiological barriers. Now we have cellular barriers. There are certain cells which would be fighting against certain things. They do not know what are they fighting against, but they definitely try to fight against certain things. So there are certain cells, macrophages is the first thing. They engulf and destroy the microbe. For example, you are the microbe. I'll just take you in and destroy you. Okay, then natural killer cells, they kill virus infected and cancerous cells. They kill virus infected and cancerous cells, but again, as it should not be specific, we should not be mentioning it over here. As we said that they are not specific. If they are not specific, then they will kill all the things, right? So let's keep it like that. Let's not go through this. You have neutrophils, which are also known as PMNL. What do you mean by polymorphonuclear leukocytes? Leukocytes as in WBCs. Leukocytes as in WBC. This is a WBC, this is a WBC, this is a WBC, everything is WBC only. That is white blood cells. There are different types. Polymorpho as in their nucleus has different morphology, my dear children. They have different lobes. Okay, they have multiple lobes, which is what is known as polymorphonuclear leukocytes. It is the first line of defense and it is highly phagocytic. Just takes it, kills. It is the first line of defense. Now monocytes, they are precursors to macrophages, helps in phagocytosis. Again, they have doing the same. So monocytes, basically from here only you get the macrophages. Monocytes is the one that will be going on further and giving rise to macrophages, my dear children, which is why it is written precursors to macrophages, and they help in phagocytosis. So whatever function it is doing, it's talking about that only. So please remember these 3 things at least. Okay. Now going ahead, cytokine barriers. What do you mean by cytokine barriers? There are certain things being released from the cells. Okay, so there are certain secretions released from the cells, and that could be, let's say, safeguarding or killing the other things. So you have a very important cytokine barrier, that is interferon. So interferons is something that is released from viral infected cells. Suppose this cell is viral infected, this will release certain interferons, and these interferons will go and tell that, hey, you have to be safe, there is a virus attack over here. So it is virus infected cells release interferons to prevent the spread of what? Infections. So cytokine barrier is that. I hope you got it. So it is said that innate immunity comes from our childhood, and it is non-specific type of immunity. Okay, just like an acidic pH of the stomach, everything that comes inside will be dying, not just virus or a bacteria. Now, differentiate between innate and acquired immunity. Let's see that. So it is inborn, it is non-specific defense mechanism, whereas when we talk about acquired immunity, it is developed after exposure to pathogens. There's a specific response to it. Okay, so it is not like I have it from childhood, I will develop it after exposure to what? The pathogens. Who are responsible for this, my dear children? So if you know, there is B lymphocytes and T lymphocytes. Lymphocytes, basically they are a type of WBC again. Now B lymphocytes, which are there in our body, they see that, okay, some attack has happened. Now this attack the foreign agents, we call them antigens. Okay, they are known as antigens. When these antigens comes in our body, what happens is that these B lymphocytes will be releasing something that is known as antibodies. Now antibodies will be fighting with this antigens, like and also the B lymphocytes will be releasing certain memory cells. So the next time the same infection comes, it knows that what type of antibodies have to be released. There are so many antibodies, but what type of antibody specifically had to be released, it knows that. Similarly, T lymphocytes also have a different mechanism of action. They also directly kill. Okay, there are different types of T lymphocytes. Some directly kill, some help others to kill, and all this thing. But you develop it after you are exposed to some pathogen. It's not like you already have it. Okay, you develop it after exposure to pathogens, and it is very specific response that it's given. As I told you, what type of antibodies have to be released also is very much specific. Now it is an immediate response, my dear children, obviously. Now over here they have to make and do a lot of things, so it is a delayed response. It takes time to what? Develop. Now specificity, it is non-specific, it acts against a wide range of pathogens, whereas it is highly specific for a particular, it works against particular pathogens in particular ways. Now is there memory? Is there any skin? Does the skin have memory? Oh, this guy tried to come inside my body. Is it like that? No, my dear children, there is no memory. So no memory does not remember previous infections, but over here, as I said, the B lymphocytes, the T lymphocytes, all of them, they release memory cells. So we have memory. It remembers the past infection and provides what? Faster responses in the future. Now, example, the components which are there is physical barrier, physiological barrier, cellular barriers, we just studied about it, and cytokines. Whereas over here, we have B cells, which helps in antibody production, and T cells, which helps in cell-mediated immunity. Cell-mediated as in the cells are going to talk among them and going to release something or going to kill the pathogen directly. That is how it works. So T cells doing cell-mediated immunity, B cells doing antibody production. Now example is skin, lysosome and everything. Here example is vaccination, immunity after infection. Once we get, for example, COVID, lots of people had COVID even without vaccine, they survived. Why? Because our body fought against it. You get it? So basically this type of immunity is acquired immunity. We are getting it. Our body is trying to recognize and try to kill it. You get it? Vaccination. Again, in vaccination, what do? In vaccination, basically you are going to give some weakened pathogens, some weakened path of the pathogen. It is not going to cause any trouble to us. For example, this is the pathogen. I am going to take a small protein out of it which is not going to cause any trouble in me, and I am going to put it inside my body, telling my body that something that carries this might come and attack you. You have to be careful. So my body produces what? Antibodies. The first time it produces, I will get fever and things like that. That happens in vaccination, right? We got fever. The second vaccination, we were fine. We were comparatively fine because now my body knows it. So it will produce the antibodies in a faster rate, or it will do its cell-mediated immunity in a faster rate, either way. So this is done after you are exposed to something. So those are the two examples that you have got over here. I hope I am able to explain you each and everything clearly. Yeah. So please write your things like that. And going ahead, draw a label diagram of an antibody molecule. I have told 10,000 times, even a 2 marker, 3 marker, 5 marker, this diagram is really important. Please try to draw it. So we have two heavy chains, my dear children. This is a heavy chain, this is a heavy chain, and we have two light chains that you see over here. Okay. Now in light chain and heavy chain, if you see, this portion is variable region. This portion is known as variable region. So this is an antibody. How many types of antibody do we have? We have IgG, IgA, IgM, IgE and IgD. Ma'am, what is Ig? Ig means immunoglobulin. Ig stands for immunoglobulin. That is antibody. Immunoglobulin as in protein. They are all proteins, my dear children. So I'll just write antibody GAMED. Gamed. I remember it like this. Gamed. Okay. So these are the different type of antibodies, and they are specific to specific type of antigens. So this region is going to be different in all of them. This is the variable region. Now towards the lower side, this is constant region, and this whole thing is also what? Constant region. This won't be changing. Now in between them, you see there are bonds. There are many sulfur bonds, disulfide bonds which are there. Over here you just see one. There's only one, but in between two heavy chains you will find multiple disulfide bonds. Over here you will be finding just one or two. Okay. Now going ahead, in the variable region you have the antigen binding site to which the antigen will be coming and binding. Okay, that is the antigen binding site. I'll give you one more information about antibodies over here. The first antibody that passes from the mother to the child through placenta is G, which is why I gave G the first thing. In game, G is the first thing. It is passed from mother to child through placenta because baby do not have any immunity. Okay, mother will be giving the ready-made antibodies to the baby so that baby can survive. Similarly, A, again from mother to child, but this time through breast milk. So which is why the mothers are advised to what? Breastfeed their children, their babies, at least during initial period of time, because this colostrum, the first milk which is released, it is very much high in immunoglobulin A. In immunoglobulin A, which will provide them immunity. So they do not have all these antibodies in the body. They have to develop it. Once something comes in, then that's when it is getting developed. But here you are providing it in a ready-made manner. What is that type of immunity where you give in ready-made manner? If you know, tell me in the comment section. Okay, I am going to talk about that, but still you can say. Question number 4 you can put and tell me the answer. Okay. So yes. Cool. And M, it is not mentioned what does do. D is not mentioned in the NCERT. What does it do? I know the function, but there is no need to tell you as of now, otherwise it will be too much confusion. IgE is allergy. In allergy, the thing that is being released in our body is IgE. In allergy. Always remember that. Okay. Cool. Now going ahead. Name 2 chemicals released by the mast cells during an allergic reaction. Mast cells. So mast cells are the one that are very important in allergy. What is allergy? Allergy is an over-exaggerated immune response. We do not need this type of response, but this is over-exaggerated, my dear children. What exactly happens in allergy? For example, here you have the mast cell, right? The mast cell will be combining with the antibodies. Which antibodies? I told you right now. Which antibodies are there for allergy? IgE. And on IgE, suppose the allergens have coming and binding. Suppose dust particle, whatever allergen is there, it is coming and binding. What will happen? This mast cell, my dear children, when this guy is active, this mast cell is going to release 2 things. One is histamine and the second one is serotonin. Both of them, what they are going to do is that, especially histamine, it is going to do vasodilation. What do you mean by that? The blood vessels which are like this, it is going to dilate the blood vessels like this, so that so many cells can come and basically try to protect the body. But actually it is a over-exaggerated response. We do not need this. We do not need this. So I saw a comment saying that some child said that I had a good voice. Okay, when I was going ahead and reading the comments, and some other said I was overacting. I feel that teacher is overacting, or probably it's a tone or accent. So I actually thought for a second that yes, probably I do a little bit of overacting. Probably it's just there. Similarly over here also they are doing overacting. So they are doing over exaggeration, my dear children. Okay, but they do not know. That it's not good for the body. But I definitely know. I actually a little bit. It's like, it's not overacting. I kind of I feel over energetic at times, and that's when it happens. But then later off, my energy goes like this. So yes, that's me. So over here, it's an over exaggerated response, my dear children, done by IgE. Please remember that, very, very important. And over here, mast cells releasing 2 things, histamine and serotonin. Good. Okay. Chalo. What is autoimmune disease? Give an example. What do you mean by autoimmune disease? That is, sometimes what happens is that, you know, okay, I'll make you learn things by relating things, okay? So basically there might be a friend in your group who is very good to you, but you do not recognize their value. You feel that they are really bad, okay? They are really bad, and you try to, exclude them from your group. Similarly, inside our body also, our body feels that something else is our own cell is a foreign particle. Our own cell is what? A foreign particle, and tries to fight back against it. It tries to what? Fight back against it. And when it does, it is going to cause harm to our body itself. And the best example for this is rheumatoid arthritis. What is it? Rheumatoid arthritis. So it is a condition which the body's immune system mistakenly attacks its own cells and tissue, considering them as what? Foreign. Example, rheumatoid arthritis. Over here in the joints, no, there is synovial fluid, my dear children. There is synovial fluid, if you know, and there is a synovial membrane. So it attacks the cells of the synovial membrane, which causes inflammation and all around the joints. It is very painful. So in this disease, the immune system attacks the joints, causing inflammation, pain and stiffness. This is rheumatoid arthritis, a very important example of autoimmune disease. Okay. Now, list any 4 common warning signs of drug and alcohol abuse in youth. Now, this is something that, okay, the reason I put this question is, we never tend to read, even I, while teaching also, I tend to feel like, no, this is not going to come anyway. So this is one such question which is there in the question bank, and you should be doing it because obviously you cannot frame it there. So my dear children, the 4 common warning signs which are there is change in the behavior, there is sudden mood swings, there is irritability, there is aggressive behavior and things like that. Second, there is decline in academic performance, there is poor concentration, lack of interest in studies and frequent absenteeism, and social withdrawal, as in you don't feel to talk to your family, your friends, you just prefer to be isolated. Okay, and the 4th thing, poor personal hygiene, neglecting grooming, messy appearance and lack of self-care. If you are someone who have all this thing, please make sure you are saying no to drugs and alcohol. Okay, there's jokes apart, my dear children. Okay, getting into this world of drugs and alcohol could be really harmful for you. Being a friend, okay, or probably being like a sister, I can tell you this, it's very, very important for you to stay away from this. Okay, there are higher chances that we keep on trying and getting addicted to this. I have got a lot of friends like that. I have got a lot of friends who after some point of time they realize that I should not have done that. You get it? And that regret is really, really sad. Okay, I agree that there are a lot of things that you should be trying in your life, but it can be something else, right? It can be the good things. Do bungee jumping. I know it is very costly. Even I have not done it yet. So yes, but still I'm telling you. But don't get into all this trouble, okay? Please. I want all my children to be in safe places. Now name a drug used as an effective sedative and a painkiller. Now this thing, sedative means you feel sleepy. Okay, and painkiller as in it kills pain. So what is that? It is morphine. It is used by doctors as well, my dear children. It is actually used by doctors during surgery and all. You know, there is critical surgeries and the people who undergo this, they have a lot of pain. So to combat that, you have morphine, my dear children. Oh, I told the answer. And then for helping patients cope with mental illness like depression, but often it is misused. What is that? For depression and insomnia. Insomnia as in you are not able to sleep. Okay. For depression and insomnia, barbiturates and amphetamines are given. Barbiturates and amphetamines are given, but they are also now being abused. They are used way too much, they are being misused. Okay. So what are the names? First is morphine, and here you have barbiturates and amphetamines. The source of barbiturates and amphetamines is not mentioned in your NCERT. But morphine, where do you get it from? You get it from Papaver somniferum. Papaver somniferum or opium poppy, my dear children. Okay. Now, mention 4 side effects of the use of anabolic steroids in males. Oh, again, a question that I tend to not give much care to, but it has come. So 4 side effects of anabolic steroids in males is that reduction in testicle size. The testis size reduces. Now if testis reduces, my dear children, it leads to what? Decreased sperm production, because sperm production happens in the testis. Second thing, breast enlargement. Males usually do not have breast enlargement, right? What do you call this? You call this gynecomastia. In Klinefelter syndrome, you might have studied this, right? So here also, because of hormonal imbalance that is being created because of the anabolic steroids, my dear children, it leads to gynecomastia. Now, increased aggressiveness and mood swings can lead to irritability, depression and also violent behavior in men. Fourth thing, liver and kidney dysfunction. Increased risk of organ damage and failure is there. One more point I added because it's there in the NCERT, that's premature baldness and enlargement of prostate. Okay, there is enlargement of prostate and there is what? Premature. Prostate is what? Prostate is a gland. You might have studied it, right? It is a gland that secretes what? Calcium and enzymes. So yes, so premature baldness and enlargement of prostate happens, my dear children. That is also a side effect of using anabolic steroids in the case of males. Now, the next thing, mention 4 side effects of the use of anabolic steroids in the case of females. First is, in the males, they were more like women-like, that's why they had breast formation and all. Here they are getting masculinized. Development of male-like features such as deepening of the voice. Second thing, irregular menstrual cycle, my dear children. This is disruption in the normal hormonal balance, which is why there will be irregular menstrual cycle. There will be excessive hair growth, increased facial and the body hair, and there will be mood swings and depression. This is common to both male and female, and enlargement of clitoris. So clitoris is enlarged over here, there the prostate is enlarged. Okay, I hope you get it. So all this thing happens in the case of females. Okay, fine. Now, differentiate between active and passive immunity, give an example for each. So the 4th question, I think it was 4th question, yeah. So basically I told you which is that immunity where we directly provide antibodies to the body. What is that immunity? It is passive immunity. So basically, active and passive, I just tell you the gist about it. Okay. Active is like being poor and then working really hard and then being rich. Okay. Passive is like already being rich, you are getting provided with all the money and everything there. You are not doing much amount of work. Okay. This is a basic thing. So in active immunity, your body is actively participating in the production of antibodies by itself. So during a normal infection that happens, for example, I am infected by anything, my body will fight back against it. It will produce antibodies. The first time it produces, it is known as primary response. It will be really slow. The second time it produces, it is known as secondary response or an anamnestic response. Okay. In that case, it will be really fast because it has the memory. So here anyway, my body is participating in production of antibodies. So this is active immunity. On the other hand, for example, a mother is breastfeeding the baby. The baby do not have anything, papa baby, right? So over here, you are providing antibodies to the baby. So is the baby doing anything to get the antibodies? Nothing. Okay, so passively they are getting the antibodies, and that is how they immunize. This is example of passive immunity. So they are ready-made antibodies that are given. So immunity developed by body in response to infection or vaccination. It can be natural infection, my dear children, or it can be vaccination also. In vaccination, you put weakened disarmed pathogens inside your body, okay, at least a part of it, and then you want the response to be created so that you have the memory for it, okay, or else normal infection that will happen. So it is produced by the immune system of the individual itself. Over here, antibodies are introduced from an external source. Sorry, I didn't read the first point. Immunity provided by ready-made antibodies from an external source, and also here, what is the antibody production? How does it work? So it is produced by the immune system of the individual, but here it is introduced as an external source, from the external source. Duration, it is long lasting and sometimes lifelong. Yes, it will be long lasting because my body itself is producing, but over there I am providing it for some time. I am providing the ready-made antibodies. It will get ended after some point of time. The money that you are taking, it will also get ended. You have to take the money again. Similarly, it will be what? Short-lived. It will be short-lived and it will be temporary, my dear children. It will be just there until the antibodies are there. Now response time, it takes time to develop as immune system has to respond. Yes, I cannot expect the antibodies will be released as such. No, here it will be giving you immediate protection. So I will give you a very important example which comes in your exams is that when someone gets a snake bite, they are given anti-venom injection by the doctor. Why? Because I cannot expect my body to release antibodies at that point of time. It will take too much time. I will die. I do not want that. That's why I am giving antivenom. Antivenom has the preformed, already formed antibodies, ready-made antibodies being there. Might be collected from someone else's body or might be made in the laboratory, my dear children. Okay, but these antibodies are ready-made and they just have to be given. Okay, so in that case, immediate protection is given. Obviously, here it is not. So antivenom and also the tetanus toxin. What do you take? You get tetanus injection, right? That also contains the antitoxins up there, and that has the antibodies, preformed antibodies. Now example is immunity developed after vaccination for polio. Yeah, after vaccination, we take vaccination for polio, right? Why? Because we want to remind our body something this can come and you have to fight against this. That is why we take vaccination for polio. Over here, immunity gained by an infant from the mother's milk, that is colostrum. And also immunity gained by the infant from the mother's placenta, that is IgE, you get IgA, IgG you get from it, right? Here you get IgA and through placenta you get IgG. Cool. Next question. Next question. Mention the names of 3 drugs which are commonly abused and write the binomial names of the plants from which these drugs are extracted. So, name of the 3 drugs, okay, which are commonly abused, as in is misused, my dear children. One is opioids, the other one is cannabinoids, and the third one is coca alkaloids. Opioids, it is taken from Papaver somniferum, that is also known as opium poppy. Cannabinoids is taken from Cannabis sativa, that is cannabis plant. Coca alkaloids are taken from Erythroxylum coca of South America, okay, of native of South America. You should remember that as well. Now listen to me carefully over here. I'll give you an understanding of this, and if you listen to me carefully, you will remember it forever. Okay, we have 3. Opioid is there. We have 2 examples, heroin and morphine. Heroin and morphine, my dear children. So heroin basically is made from deacetylation of morphine. Okay, there's a chemical modification being done and that's how you get heroin. So morphine acts as a sedative and a painkiller. Heroin which is there, it is a depressant. It causes depression. It is a depressant as in it slows down your body very much. Okay, it is a depressant. Heroin is a depressant, morphine is not. Now, where do you get this from? Sorry, morphine is a sedative and a painkiller. Okay. Now, where do you get it from? You get it from the latex of opium poppy, the milk of opium poppy, the opium, the pods, no, in the pods, there will be milk, from that you get this. The picture is given of the plant, my dear children, you should recognize the plant also. The question can be coming from that aspect as well. I think I have done a 2 marker, 3 marker, 5 marker. If not, please go through it once. Okay. Now, where do they affect? Opioids, where do they affect? They affect the central nervous system, CNS, that is the brain and spinal cord, and also gastrointestinal tract. Gastrointestinal tract. It is also known as smack. It is also known as, heroin is also known as smack, my dear children. Cool. Now we have cannabinoids. Cannabinoids, you get it from cannabis sativa plant. Okay, there are different parts of the plant, the flower, the leaf, everything can be utilized to form different type of drugs, that is marijuana, hashish, ganja, bhang and everything. Where does this guy affect? This guy affects the cardiovascular system. This guy affects the cardiovascular system, as in your blood vessels, your blood and everything up there. Okay, it affects your heart and the blood vessels and everything. Now, please remember, nicotine is something different, cannabinoids is something different. In your book, the question comes, who increases the blood pressure and things like that? It is nicotine. Okay, cardiovascular system, it is mentioned, then it is cannabinoid. Cool. Okay, the structure of cannabinoid is given where it has something like this. Okay. Not like this. Okay, chemistry ma'am, Aishwarya ma'am will kill me if I make like this. So there is a bridge like this in their structure, so that is a cannabinoid. Okay, in the case of, they have given a structure for morphine also, so it looks like an O, so I remember that it comes from opioids. Okay, I'll show you if I have the picture, otherwise you see it from the 2 marker, 3 marker, 5 marker. Okay, so this is cardiovascular system. Now what about coca alkaloids? Cocaine. Okay, cocaine is also known as coke or crack. Cocaine is also known as coke or crack, my dear children. It is from Erythrocylum coca, as I told you, South America, and basically it affects the CNS again. Okay, in CNS particularly, they are going to mess up around dopamine receptors. Okay, dopamine receptors. So from one neuron to another neuron, some neurotransmitters are released, that is dopamine. Now they are continuously released, they are not taken back, they are not taken, but they continuously release, which is why you get a sense of euphoria, you will be so happy. Okay, there will be hallucination, you will see cows flying and things like that. You might be thinking that, ma'am, we are seeing now also. So it's not like that. Okay, this is completely different. This is hallucination, is very much dangerous. Not as fun as I'm talking as of now. Okay, you might be crossing the road, but you feel that you are flying. What do you say? Vehicles might be coming. You might not know about it because you're hallucinating. It's very dangerous. You get it? That is known as this, and it causes what? Hallucination. It causes hallucination. Apart from them, there are 2 other plants also given in your NCERT that causes hallucination. One is Atropa belladonna and second one is Datura. Atropa belladonna and second one is Datura. Both of them also cause hallucination along with this guy. Okay, and the picture of Datura is also given in your NCERT. Go through it. Done? Done? Are we done? Yes, we are done. Next, tobacco smoking associated with rise in blood pressure and emphysema substantiate. So I told you about nicotine, right? So tobacco, it contains something that is known as nicotine, and nicotine, basically what does it do? It stimulates our adrenal gland, which is there on our kidneys. Okay, it stimulates our adrenal gland to release adrenaline. Adrenaline is a fight or flight hormone. It makes it highly energetic, you know. We have to either fight or we have to run away. So adrenaline increases the heart rate and blood pressure. So this is the one that causes increase in blood pressure, my dear children, and it leads to hypertension. Now that is one reason how it causes that. Okay, because nicotine is stimulating the adrenal glands to release adrenaline. Talking about the next thing, that is emphysema. Emphysema is basically a respiratory disorder. Now how do we get emphysema? Tobacco smoke increases the carbon monoxide content in our body, and you know what, usually the hemoglobin loves oxygen, but when it sees carbon monoxide, it's like, I don't know whether I love oxygen or not. So it goes and binds with carbon monoxide, my dear children, and hence our body would not be getting enough amount of oxygen. That's a problem. So carbon monoxide, which reduces the oxygen carrying capacity of hemoglobin. It reduces the oxygen carrying capacity of hemoglobin. Smoking damages alveoli in the lung, my dear children, reducing their elasticity and surface area for gas exchange, and this leads to emphysema, causing difficulty in breathing and oxygen deficiency in body. So tobacco smoke increases the carbon monoxide content. It basically competes competes with hemoglobin to bind with its oxygen carrying capacity decreases. Apart from that, the smoke will also cause damage in the alveoli. If alveoli is not there, gaseous exchange cannot take place, and this will finally lead to difficulty in breathing and things like that, which occurs in emphysema. Okay, works. So depression and insomnia is, tell me the answers, tell me in the comment section. Depression and insomnia is caused by which drug? A and B. A and B. A and B, what is it? Amphetamines and barbiturates. In between, I'll keep on asking, you have to tell me. Okay, going ahead. Yeah, this is the bridge I was talking about. Okay, so the skeletal structure of a drug is shown below. Answer the following question. Identify the drug. It is what? Cannabinoid drug. And name the plant source of this is cannabis sativa. How is this drug generally taken? How it is taken also, that is also a question. Generally taken by inhalation or oral ingestion. Okay, oral ingestion or inhalation as well. You can inhale it or you can take it like that. Okay, and what was I saying? I was saying something. Yes, there's a sentence in your NCERT which says that cannabinoids are abused by sportsmen. So what is that drug that is abused by sportsmen? It is cannabinoids. Please remember that also. Children, I hope you're not getting bored. I'm trying my maximum to be highly energetic. You know, it needs a lot of energy to be energetic like this, because if you're there in front, it feels obviously you and me both are talking, but only over here I am the only one who is talking. So you need to pace up that energy. Yes. Now fill the missing data in the table depicting diseases, their causative agents and the symptoms. Very important question. So I've just given you a gist of it, but you have to remember all the pathogens, what do they cause, what is the disease that they cause, what are the symptoms that is there, do they have a vector, do they have a test, okay, not test, do they have a test for analyzing if a person has the disease or not. Okay, all this information you should know. So I have just put it up there. The answer is this. Ascariasis is caused by Ascaris, and it causes, the symptoms of it is what? So Ascaris basically goes and affects our intestine, right? So it is like noodly thing on an intestine, and if it's there, obviously it will cause abdominal pain. It will lead to internal bleeding as well, my dear children. It will lead to muscle pain, fever, anemia, blockage of the intestinal passage, not package, okay, passage. The second thing, ringworm. So trichophyton, epidermophyton and microsporum. Okay, microsporum, trichophyton, epidermophyton, all of them are going to cause a disease that is known as ringworm disease. Now all of them are fungus. Do you know that? They are all fungus which are causing what? Ringworm disease. Ringworm, it is known as ringworm because there is a ring shaped thing that comes and it looks like a worm. So it is not a worm, it is caused by fungus. And what are the symptoms? Appearance of scaly lesions on various parts of the body and all. Okay, now typhoid is caused by Salmonella typhi. It is a bacteria. What is Ascaris? Ascaris is a helminth. Aschelminth, it's a worm. Okay, Ascaris and Wuchereria, both of them are Aschelminth. Now Salmonella typhi is a bacteria. It causes high fever, it causes weakness, it causes headache, it causes stomach pain, it causes constipation. Okay, lots of questions can come around it. What is the extreme thing that happens is that intestinal perforation happens, the person could die. What is the test? Okay, so in your book only two diseases are there for which you have test. One is typhoid, other is HIV. For typhoid the test is what? Vidal test. Vidal test. Okay, and how it is spread? Through feco-oral route, that is through fecus. It is contaminating the water, the food, and the food if we eat or the water if we drink, we get the disease. Okay, so you should know the route also. Pneumonia is caused by Streptococcus pneumoniae. It is a bacteria again, my dear children. Fever, chills, cough, headache, difficulty in breathing, everything happens over here. Please remember, in the case of pneumonia and everything, it is going to mostly affect your whole respiratory system, basically majorly your lower respiratory system. When you talk about common cold also have this thing, nasal congestion and everything is there, but please remember it is not going to affect your lungs. It is not going to affect your lower respiratory system. It affects the upper one. So nasal congestion and discharge and sore throat and cough and headache is found in common cold, and it is caused by rhinoviruses. Okay, now again disease related question that can come is, as I said, the roots of how diseases are transferred, that is there. And then I was going to say something, I forgot. Yeah, which are those diseases where you need vectors? Okay, so you have dengue, And chikungunya, it is spread by Aedes mosquito, my dear children. It is spread by the bite of Aedes mosquito. Malaria, you know, it is caused by female Anopheles mosquito. There is filariasis or elephantiasis. It is caused by the bite of Culex mosquito. Culex mosquito. About this I have discussed in 2 marker, 3 marker, 5 marker, but still telling you if at all you have forgotten. Okay. Going ahead, we have schematically represent the mode of infection in HIV. Let me see how many questions are left in this particular chapter. Okay, I think, where is it? Okay, we are here. Okay, schematically represent the mode of infection in HIV. How does it happen? Let's see. So, my dear children, in HIV, this is the HIV virus. Okay, you have a capsid that was formed of what? Protein. Okay, and apart from that, inside that they have the genetic material which is a single-stranded RNA. Single-stranded RNA, they have 2 of them, but they are not bounded to each other. They are 2 separate ones. You don't have to write 2, 3 and all. It will confuse the teacher and also you. Just write it is single-stranded. There is no complementary strand to it. Okay. Now, this virus will slowly go affect the animal cell, and mostly it is first going to affect the macrophages in the body. Macrophages, basically they have the ability to take things inside, right? So, but it's not like that. There are certain receptors onto which these guys bind, and slowly what will they do is that they insert their genetic material. Please, this question is very much important. Previous year it has come. Okay, I want you to pay attention to this. Okay, anything around it could also be coming. So, you have got the viral RNA inside. Now, what happens is that this is RNA. What we have in our body, we have DNA as a genetic material, my dear. We also have RNA, but RNA is not a genetic material. What is a genetic material? It is a DNA. So, this guy, the RNA, will be transcribing back into DNA. What, ma'am? DNA transcribes to RNA. RNA does not transcribe to DNA, right? No, baba. So, usually it's a DNA that will be transcribed to RNA, but over here reverse transcription happens. Opposite of it happens. So, RNA is going to make the DNA by the help of a very important enzyme, that is reverse transcriptase. It reverses the process of transcription. Now we have DNA. This viral DNA incorporates into the host genome, my dear children. It will get into the host genome, that is our genome, and then our machinery, everything will be used. New viral RNA is produced by the infected cells. A new viral RNA will be produced. Right, new viral RNA is produced, and then obviously this RNA will undergo translation, everything. It will make proteins that is needed. For example, this enzyme is a protein. So, all these things are being made. So, new viral RNA is produced from the infected cells. This mRNA that comes around, my dear children, will give rise to what? Information for making more proteins, and information for making the capsid, and obviously you get single-stranded RNA also. This can be incorporated inside, and each of them, now new viruses would be produced outside, and these viruses are going to infect other cells. Always remember, the first cells that it affects is macrophages. Okay, it affects macrophages, and macrophages is where there are a lot of HIV viruses being born. Now, slowly they are going to infect. They are going to infect their ultimate motive, that is T lymphocytes. So, T lymphocytes is the one that is doing what? Cell mediated immunity. So, it informs the other cells to basically help in the production of, it informs the B cells to produce antibodies, or probably it goes and tells other cells to produce something else, which helps in the whole thing. So, they are like they have a helping mentality, which is why I call them T helper cells. So, you have 2 types of T cells. T cytotoxic cell and T helper cells. 2H cells are the one that is primarily affected, my dear children. Here they are made, and then they HIV factories, they are known as HIV factories, and from here they go to TH lymphocytes, they kill off all of them, they damage all of them, and that's why our immune system goes down, weakens. So please, all the steps over here, what is happening in each step, you should be knowing it. Okay, going ahead. A person has been diagnosed as HIV positive. Name the diagnostic test which the person underwent. What is the name of the test, my dear children? It is ELISA, enzyme linked immunosorbent assay. I told you there are only 2 diseases which have a test. One is this HIV, and the other one is what do you call it? Vidal test, that is for typhoid. Okay, so ELISA stands for enzyme linked immunosorbent assay. It works on antigen antibody principle. It works on antigen antibody principle. I think I taught you in some chapter. Anyways, should I tell that over here? Do you need antigen antibody? Children, if you need antigen antibody principle, tell me. I'll take it in the MCQ session tomorrow, okay, if you need it. Now write full name of the pathogen involved. HIV full form is human immunodeficiency virus. It is making us deficient, immunodeficient, right? So human immunodeficiency virus. Now name the genome of the pathogen. Baba, it's what? Single stranded RNA, ssRNA, single stranded RNA. Now which particular cells of this person are likely to get destroyed? It is what? T helper lymphocytes or helper T lymphocytes. Okay, you can just write this, you don't need to write this. Helper T lymphocytes or T helper lymphocytes, either way. Okay, now suggest the partially effective treatment to him. What is the partially effective treatment? It's not completely curative. Do you know that HIV is not completely curative? Yes, it is not. So what is a partially effective treatment which lets them survive? It is antiretroviral therapy, because HIV is a retrovirus. That is why antiretroviral therapy is given to them. Cool? Works? Now, it's all about cancer. Okay, I have included some more questions for cancer, because cancer is a very important topic. Now, name the group of genes that have been identified in normal cells that could lead to cancer, my dear children. So, group of genes that are there normally in our cells, my dear children, they are known as proto-oncogenes. They are known as proto-oncogenes. These proto-oncogenes do their regular work. But if they are mutated, it will lead to cancer. It will lead to cancer. So, proto-oncogene. So, group of genes that could lead to cancer is proto-oncogenes in our body. Now, name the technique that are useful in detecting cancers of internal organs. For internal organs, what do you need, my dear children? You can have MRI, magnetic resonance imaging, or also CT scan. Computed tomography, over here you use X-rays. Over here you use magnetic radiation. Okay, so so magnetic resonance imaging and also CT scan, my dear children, that is computed tomography, is the one that you use for detecting cancer in the internal organs. Now, why are cancer patients often give alpha interferon as a part of treatment? Ma'am, you said interferons are released from virus infected cells. That is different interferons. Okay, so I agree, those interferons are released from virus infected cells and they tell the other cells that you have to be safe. This is alpha interferons which is given to our body. For example, suppose someone has cancer. They are given alpha interferons because they are immunologically very down. You know, they have so many therapies, chemotherapy, this therapy, that therapy. So in that, we target the cancerous cells, but also the normal cells die. So there are certain good cells also which die in this process, which is why we need to boost up their immunity. Okay, so that is why alpha interferon is given. It activates the immune system and helps destroy cancerous cells. It boosts the body's natural defense mechanisms to slow down what? The tumor growth which is there. Cool? Next, stages in the life cycle of plasmodiums. Plasmodium is given below. Identify A, B and C, D. I saw this question and I was like, okay, so what is this? What is happening over here? So identify the stages. If you are asking about the stages of the plasmodium, then you have to go a bit further. So my dear children, from the liver, okay, in the liver, the cells, they are affected and later they open up things. What are this? They are in the sporozoite stage. They are in the sporozoite. Where is it? Sporozoite stage. Infectious stage injected by the mosquito. Okay, that is A. Where? Over here. Not here, over here. Okay, that is what I was thinking about. So as I told you, in the salivary gland you have sporozoite. This is injected into the human body. It goes to the liver. In the liver they are going to transform into the schizont stage. Okay, schizont stage, liver stage, where the parasite multiplies. Now next stage is C, where basically what is happening? It is going and infecting the other cells as well. That is the RBC, my dear children. That is the RBC. So it is merozoite stage. So it is basically released into the bloodstream, infecting the RBC. So over here in the schizont stage, it is multiplying, right? And the merozoite stage, it is released, and now it is getting into the RBC, my dear children. In the RBC, what they are going to do? They are going to form gametocyte stage. That is not mentioned over here, that I am not answering also. But now D is given over here. What happens? What is this? So basically the human host, it got completed over here. The gametocytes are there. The gametocytes are being taken up by the mosquito. In the mosquito, the gametocytes are going to fuse. Okay, sorry, the gametes are going to fuse and give ookinete. And give what? Ookinete, which will later transform into what? Sporozoite. Which will later transform into sporozoite. I don't think this question would be coming. because this much information is not there in your NCERT, but still let's be prepared. So in salivary gland, sporozoite stage. In liver, when it reaches and multiplies, I call the stage as schizont stage. Later when it goes and infects RBC, the stage when it's going and infecting the RBC, I call it merozoite stage. Okay, I call it merozoite stage. In the RBC, it is gametocyte stage. Now these are gametocytes are taken to the mosquito's gut. In the mosquito's gut, they form the gametes. The gametes are going to fuse there. Okay, the gametes basically are going to fuse there and form ookinet, and form what? Ookinet, which will later transform into sporozoite. Fine. Now name the toxin responsible for appearance of symptoms of malaria in humans. What is that toxin? I told you, what is the symptom? It's fever, high fever and chills, right? So what is the toxin released responsible for it? It is hemozoin. So it is a toxic substance released when infected RBCs rupture, causing fever and chills in the malaria patients. Okay. Next, the flowchart shows replication of the retrovirus in the host. Fill in the missing data in the boxes labeled 1 and 2. So let's see, this is 1 and this is 2. So retrovirus is there, virus infects the normal cell, viral RNA is introduced into the host cell. Now reverse transcription happens. When reverse transcription happens, what do you get? You get a complementary DNA, or you get a DNA. You say that you get a DNA, okay, either way. So see, a DNA or complementary DNA formation occurs. Now viral genetic material in nucleus of the host cell, yes, it is put into the nucleus of the host cell. The viral DNA incorporates into the host genome. It will incorporate into the host genome, my dear children. And then what happens? Then what happens over here? The virus, what will it do? Transcription and translation of the viral proteins and everything will happen. Okay, it will form the capsid and everything. The virus comes out of the host cell. New viruses can infect the other cells. So box 1, you can just say DNA formation occurs. Don't mention complementary DNA formation. You can just say DNA formation occurs. Okay. Next thing over here, you can say that viral DNA incorporated into the host genome. Okay. And here transcription translation happens. Here you can also mention that viral RNA and the proteins are being made. Okay, viral RNA and the proteins are being made, and then it is transferred. That's all. So transcription and translation of viral proteins, okay, happens. So how do you write it? Here basically you can write it as this also: transcription and translation of viral proteins, and also viral RNA is made. That's all. In that way you are safe. In that way you are safe. Just for being safe, I have told you that. Now, give an example for the retrovirus. Name the give an example for retrovirus. What is the example? HIV. HIV is a retrovirus. Now, name the disease caused by this retrovirus. It is AIDS. Acquired immunodeficiency syndrome. What do you mean by that? You are acquiring this, and this is making you immunodeficient. And when you become immunodeficient, you have lots and lots of symptoms. That is syndrome. Syndrome is a group of symptoms. So acquired immunodeficiency syndrome. Now you have named the diagnostic test for this disease. What is the diagnostic test? It is ELISA, enzyme-linked immunosorbent assay. Cool? Works? Done. Next, observe the diagram and answer the questions given below. Identify A. What is A, my dear children? A is lymph nodes. A is lymph nodes. Let's identify first, okay? What is B? B is here. It's like middle, right? It is, B is thymus. What is C? C is basically lymphatic vessels. Listen to me carefully. We have lymphoid organs. We have two lymphoid organs with two types of lymphoid organs, primary and secondary. Primary lymphoid organs are where the lymphocytes are being formed, okay, they are produced, they mature, and they become antigen sensitive. They become sensitive to antigen. They know that these guys, there are foreign particles we need to kill, but they don't kill there. They just mature, they produce, they mature, and they get antigen sensitive. Okay. Now, where do they actually go and do the action? It is in the secondary lymphoid organs. So, in primary lymphoid organ where they actually form and everything, you have two things, bone marrow and thymus. Bone marrow and thymus. So, this is what, not secondary, if I said secondary, I am sorry. The primary lymphoid organs, basically you have two where they actually form and all. It is thymus and bone marrow. So, this is primary lymphoid organs. And lymph nodes is a secondary lymphoid organ. This is where they go and do the action. Okay. Now, bone marrow is the place where you have hematopoietic stem cells. You have certain stem cells which have the ability to give rise to lots of blood cells. And one such blood cell is WBC, and one such WBC is lymphocyte. You have B and T lymphocytes forming in the bone marrow itself. So, bone marrow is a place where everything is formed. Now, thymus is where the T lymphocytes, exclusively the T lymphocytes will be coming over here and getting matured in the thymus. Fine. Now, they are going to go to different locations of our body. One such location is the lymph node. Where do you have the lymph node? So, basically what happens is that, you know, our normal blood vessels, we have capillaries, and so arteries opens into capillaries, capillaries opens into veins. So, capillaries are very thin things, and there are high chances they have small pores, so blood and lots of fluid can be moving out of them. Yeah, lot of fluid can be moving out of them. And when they do, when these things move out, basically what happens is that, okay, when the things move out, basically what happens is that there should be someone to collect all this fluid. Otherwise, basically we are losing a lot of things, and it causes inflammation as well at that particular location. So, there you have lymphatic vessels. So, lymphatic vessels will reach out to areas, will take in all the fluids in there. This fluid is basically from our blood only, okay, the plasma and certain cells. It will also have certain pathogens which are trapped in our blood. All this thing is taken by the lymphatic vessels. The lymphatic vessels, they look like this. They form small areas like this. Over here, these areas are known as lymph nodes. They are known as lymph nodes. So, here also you can see these are vessels, and here they have nodes elsewhere. These are what? Lymph nodes, my dear children. And lymph nodes have so many lymphocytes. And lymphocytes, when they see the pathogen, they are gonna kill them. I told you this is a site where they do the distribution. Get it? I hope you get it. Apart from lymph nodes, you have spleen, which is above the stomach, my dear children. And then you have MALT, mucosa associated lymphoid tissue. Mucosa is the innermost layer of our gastrointestinal tract. So, associating to it, you have tonsils and you have Peyer's patch in your intestine. In the intestine, you have Peyer's patch. Over here, you have tonsils. All of them are areas where distribution is happening. So, spleen, lymph node, MALT becomes a part of secondary lymphoid organs. Okay. So, lymph nodes filter lymph, trap pathogens, and activate the immune response by producing what? Lymphocytes. Now, B is what? Thymus. Thymus is responsible for maturation of T lymphocytes, and they are crucial for cell mediated immunity. They are crucial for what? Cell mediated immunity. You know that, right? Now, lymphatic vessels, you don't have to write the function, but still have written. They transport the lymph as in the fluid, okay, throughout the body, helping the drainage of excess tissue fluid and the circulation of immune cells as well. Done? Done. Now, these are the questions that I added extra because I wanted to give you a more gist about cancer cell. I told you cancer is a very important topic. The important topics, I might cover it once more. So, characters of cancer cell, you should be knowing about that. So, they do not show contact inhibition. Please remember that. What do you mean by contact inhibition? For example, I am a cell. Okay, you might have seen people who, like, even if there are lots of people already in the room, they'll move like this. Similarly, cancer cells are also like that. So, basically when the normal cells divide and they come in contact, they stop the division. But cancer cell, they keep on dividing. That is known as loss of contact inhibition. So, usually we get inhibited by contact, but they have lost that property. This is loss of contact inhibition happening over there. Okay. So, let us see the points. So, cancer cells lose contact inhibition, meaning they continue to divide uncontrollably. So, they have uncontrolled growth. You can write that in separate points also. They have uncontrolled growth, they lose contact inhibition. They form tumors. They form tumors, my dear children. And these tumors can be benign as in it stays at one place, and malignant as in it spreads. The feature of malignant tumor is that the cells which are involved in malignant tumor is that they show a process which is known as metastasis. Metastasis means that they can spread to different parts of our body. So, malignant tumor, whenever you say you have a malignant tumor, that means the cells are actually doing metastasis. They are moving around different body parts and they are spreading. Benign stays, you can remove that also. Okay. The malignant tumor invades surrounding tissues and compete for nutrient. This is also a property. So, they lose contact inhibition, they divide uncontrollably, they form tumors, it can be benign and malignant. Some of it they show metastasis, okay, that is they move around different body parts spreading it, and basically they invade the surrounding tissue and fight for nutrients as well, they compete for nutrients. Now cancer causing agents are known as carcinogens. You have physical agents, that is ionizing radiation, that is X-rays, gamma rays. Now non-ionizing radiation like what? UV rays. UV rays are non-ionizing. Both of them are physical agents. When these agents put up on our DNA might have some mutation, which is why there is a cancer being formed. Now chemical agents include tobacco smoke, which leads to what? Lung cancer. Biological agents are oncogenic viruses. There are certain viruses which affects our body and causes cancer in us. Okay, they are known as oncogenic viruses. Okay, and they contain viral oncogenes. They have certain genes, they put it in our body and then we have cancer. Cellular oncogenes is also part of biological agents only. So in our body, I told you there are cellular oncogenes and proto-oncogenes which are doing their own function. But basically if there is some mutation, it can lead to what? Cancer. So it is not like they can be activated and all. Basically some mutation in them can lead to cancer. Now diagnostic tool, you have biopsy and histopathological study. Biopsy means you are cutting a tissue part and then you are seeing it. When you are studying the part, you call it histopathological studies. So microscopic examination of stained tissue samples is biopsy and histopathology. Biopsy means removing that and seeing it, okay. And now removal of that thing is known as biopsy. Studying about that is histopathological study. Now you have blood and bone marrow test, detect increased cell counts in leukemia, so that is also a thing. Yeah. Now you have radiography, that is by X-rays, CT scan, computed tomography and MRI, which is used for detecting what? Internal cancers which is there. Now antibodies can also be checked. For example, a person who has cancer, we know that they produce these type of antibodies, and if you find the same type of antibodies in someone else, we get to know that yes, probably they are also having cancer. Now molecular biological techniques to identify inherited cancer susceptibility genes is also there. Some people have that cat of getting there are chances of them to inherit cancer, so that cannot be removed, that probability also cannot be removed. If molecular biology is actually working on that, so that they can find the genes and basically look at that, okay, this person has a risk of getting cancer. Okay. Actually, so we completed one chapter. Now we are getting into microbes in human welfare. From this point onwards, it will be a bit faster than the other one, because the first chapter, as I always say, it takes a bit of time because they are high weightage chapter which I have put in first, so that you don't miss. So you know what happens is that they get bored, children get bored and they go off. They don't see the entire video, so which is why I have put the priority chapters on the first thing. It does not mean that you should be missing out on the other chapters. They are equally important. All 1, 2, 3, 4 mark, everything, every mark which you are having, my dear children, is very, very important for you. Now let me just take a break. Let me just drink a little amount of water, okay. You also take a 2-minute break and come fast, okay. Okay, are we back to the battleground? Let's begin. Yes. So we have microbes in human welfare. That is the next chapter. I love this chapter a lot. I don't know why, but I like it. So yes, so discuss the role of microbes in household products. Household products, what all microbes do you use? Have you seen that your mother or father, basically they add curd into milk? What is that? Basically, this particular curd which is there, it has a bacteria that is known as lactobacillus, and when it is put into the milk, again it will convert the milk, it will basically break down the casein and things like that. It would make it in a different format. It's not breaking down the casein, it actually feeds on the protein that is known as lactose and convert it into lactic acid. In the milk there is lactose, it converts into lactic acid, and when there is lactic acid, what happens is that the milk protein, that is casein, it changes shape and things like that, and basically it clumps up forming the curd. So first thing is lactic acid bacteria grown in milk, okay, and it converts it into curd. LAB produces acids that coagulate and partially digest, okay, partially digest the milk protein, that is casein. A small amount of curd added to fresh milk as inoculums or starter is added. Now one very important thing, the lactic acid bacteria basically increases the amount of vitamin B12, which is why we have said that we should be having curd, because curd have a high amount of vitamin B12. Okay, now again, baker's yeast, you might have seen people using yeast, right, for making cake, for breads and things like that. So baker's yeast is what? Saccharomyces cerevisiae, my dear children, and is used for making the bread. Yeah. Now toddy, a traditional drink, is made by fermentation of sap from the palms, which is actually not a household product, but still. So it is a traditional drink. It is made from fermentation of sap from the palms. Now, you know Swiss cheese, the one that I always say, give this example, don't kill me for that. So basically in Jerry, Tom and Jerry, the Jerry used to sneak out in search of the cheese, right? What was it? The cheese used to look like this, which has large holes, my dear children. It had large holes. So basically this is Swiss cheese. This is what? Swiss cheese. And why there are large holes? Because there is a bacteria that is Propionibacterium shermani, okay, which basically releases a lot of carbon dioxide. When it releases carbon dioxide, there are big holes being left behind. So large holes in Swiss cheese are due to production of large amount of CO2 by a bacterium that is known as Propionibacterium shermani. And then you have Roquefort cheese. Roquefort cheese is very expensive, okay. Roquefort cheese is ripened, actually it is ripened by a fungi. A fungi that is known as Penicillium roqueforti. If you do not want to remember it, please don't remember it. It's not there in your NCERT anyways. It's not there in your NCERT, don't remember it. Otherwise, you will confuse with Penicillium rotatum and Penicillium roqueforti. I don't want that. So, specific fungus is there which does that. You remember that only. Works. So, lactic acid bacteria, baker's yeast, that is Saccharomyces cerevisiae. Now, this Saccharomyces cerevisiae can be brewer's yeast also. It can be used in brewery also. Okay. Now, toddy, the large holes in Swiss cheese and the Roquefort cheese, all of this are really important. Going ahead with the next thing, explain the roles of microbes in industrial products. In industries, how they can be used, basically for production of acids. Okay, so some are very easy to remember. For example, acetic acid. Acetic acid is by Acetobacter aceti. Name is same. Acetobacter aceti produces acetic acid. Clostridium butylicum produces butyric acid. Now, the thing that might be a bit odd to remember is Aspergillus niger. It produces citric acid. Aspergillus niger, my dear children, it's a fungus and it produces what? Citric acid. Please remember. Acetobacter aceti and Clostridium butylicum and also Lactobacillus produces lactic acid. They are all bacteria. They are all bacteria, but this guy is a fungus. Now, yeast, Saccharomyces cerevisiae, it produces ethanol as well. I told you alcohol production is there. So, it produces ethanol as well. Now, Penicillium notatum, which is a fungus, okay, which is a fungus, it produces penicillin, which is an antibiotic. Again, it can be used in the medical industry, right? So, it is a industrial product. Now, Streptococcus is a bacteria that produces streptokinase. What does streptokinase do? It is a clot buster. It bursts our clot. So, no one is going to have any clot. So, streptokinase is given to burst the clots which is there in the blood vessels and all. So, people who have myocardial infarction, heart attack and all, they just given this particular thing so that the clot is being removed. Now, Trichoderma polysporum, which is a fungi. It is a fungi and this fungi produces cyclosporin A. What does this cyclosporin A do, my dear children? Cyclosporin A is used as an immunosuppressant agent. It suppresses the immunity. You might think until now we were talking in cancer and all, we want to boost our immunity. Why are we suppressing our immunity? So, for example, I have a transplantation. Now, in transplantation, obviously, I am taking a foreign thing, so my body will kind of react to it very vigorously. I do not want that to happen. So, in that case, I have to take an immunosuppressant. So, cyclosporin A, which is produced by Trichoderma polysporum, which is a fungus, is an immunosuppressant. Then you have Monascus purpureus, which is producing statins, my dear children. Statins basically is a blood cholesterol lowering agent. It lowers the blood cholesterol. Please, blood cholesterol. Okay. Blood cholesterol is being lowered by that. So, Aspergillus niger, citric acid, Acetobacter aceti, acetic acid, Clostridium butylicum, butyric acid, Lactobacillus, lactic acid. 3 of them are bacteria, 1 of them is fungi. Going ahead, yeast Saccharomyces cerevisiae, fungus again. This is a fungus that produces ethanol, and Penicillium notatum, again a fungus that produces penicillin, but this penicillin was antibiotic, does not allow the growth of another bacteria. Streptococcus, a bacteria that releases streptokinase, which is a clot buster. Trichoderma polysporum, which is producing cyclosporin A, which is an immunosuppressant agent. Now, we have Monascus purpureus, which produces statin, and statin is a blood cholesterol lowering agent. Works? Chalo. Next thing. Explain the process of sewage treatment. So, sewage treatment I have to explain like this, then we will explain it in a different way. Shall we begin? So, listen to me. First, we have primary treatment. And then we have secondary treatment. I am not writing the whole thing. Okay, now listen to me. I have to only listen to me, right? Okay, so what happens is that things which can be easily filtered out, as in things which will be floating, I can remove it out with the help of a process that is known as filtration. Second thing, things which are very big, they will be sedimenting down, so I can be removing those with the help of sedimentation. So both these steps comes under what? Primary treatment of the sewage. Now what are we left with? The fluid and everything that is left with is known as primary effluent. You have to imagine, okay? Now I am going to put this primary effluent into big tanks. What tanks, my dear children? Aeration tank. That means there is lot of air over here. And when I put the primary effluent over here, what happens is that this primary effluent will be having so much amount of organic matter. And seeing this and the aeration tank, what is going to grow up? It is the aerobic bacteria. Aerobic bacteria, my dear children, it will be growing really up. And when they grow up, they are going to basically break down this organic matter. But for them to work, you constantly need what? Oxygen. They are aerobic bacteria, so they need oxygen. And what do you call this process as? What do you call this thing as? BOD, biochemical oxygen demand. So it's the aerobic bacteria which demands for oxygen for breakdown of organic matter. So if I say the BOD is too much, that means there is too much amount of organic matter, which is why aerobic bacteria is needing too much amount of oxygen as well. If I say BOD is less, that means what? Organic matter is less, that is why aerobic bacteria which is there, it is going to utilize less amount of oxygen as well. So we have to work until BOD is less. It works until the amount of BOD is reduced. BOD is the oxygen required by the aerobic bacteria to break down the organic matter in 1 liter of water. So if BOD is way too much, that means there is high amount of oxygen, that means organic matter is way too much, and that means it is there is lots of what? Pollution. The water is highly polluted. If this is less, until that we'll go on, we'll keep on doing it. And listen to me, while it does it, aerobic bacteria breaks down the organic matter, it kinds of become friends with the fungal filaments, and everything. And this combination of aerobic bacteria plus fungal filaments plus the extracellular things that they release will be now known as flux. This is a mesh-like thing that is being formed. This mesh-like thing is the one that is targeting the organic matter. And then slowly they will keep on working in the aeration tank, and what happens? The BOD will actually go down. And at this point of time, this whole thing which is there, it is transferred to a next thing which is known as a settling tank. It is the secondly settling tank where it is transferred. Now in the settling tank, what will happen? The flux and everything will be arranging down. Okay, and the fluid and everything which is clear of organic matter is above. This fluid can be removed down into streams, rivers, or whatever you want because it's totally clean. What is left behind is the flux, right? But now you'll call this as activated sludge. What do you call it? You call this as activated sludge, my dear children. A little amount of activated sludge, we'll take it and we are going to put it over here. Why? Because the next set of primary effluent is going to come, so you need some bacteria. So it acts like an inoculum. So it will act like an inoculum. The rest which is left behind, okay, the rest which is left behind is being taken to the next thing that is known as anaerobic sludge digester. That means when there is no oxygen at this point of time, they are going to work. Without oxygen, there are some bacteria which is going to work. And what are they going to work on to? They are going to work on to this activated sludge. This anaerobic sludge digester, in this we put anaerobic bacteria, and this anaerobic bacteria is going to feed on all this thing and produce what? The biogas. Produce the biogas. What does the biogas contain of? It contains of methane, it contains of H2S, it contains of CO2, my dear children. All of them come together to form the biogas. It is being produced. So with the help of sewage treatment, I get clean water, and also I get biogas which can be used as gas in my kitchen or can be used for electricity purpose as well. That's so great, right? So the anaerobic bacteria which are used over here, they are methanogens, okay, because they are producing methane. Get it? I hope you got it. So this is the whole treatment about. If you want to read, you can read it as well. Okay, that particular slide is gone. I hope you know that. But if you read this, now you will understand it clearly as well. So you can read it. Okay. I'm not going to read over here because it might take too much of time. It's long. Now, describe the role of microbes as biocontrol agents and biofertilizers. So biocontrol agents, what do you mean by that? As in, I put pesticides, right, to kill the pest. But I don't want to put pesticide because it is kind of very toxic, right? I will be taking this pest inside, I'll be taking these chemicals inside my body. I don't want to do that. So basically, I can use biocontrol agents instead, which are going to eat my pest, certain organisms which actually feeds on pests. So I don't need the pesticides, right? So for that, I have few things. Let's see who are those. First thing, biocontrol agent, what do you mean? So it will greatly reduce our dependence on toxic chemicals and the pesticides, my dear children. Right? Now, the ladybird, a familiar beetle with red and black markings, and the dragonfly are useful to get rid of what? Aphids and mosquitoes. So, over here, ladybird, which is a beetle, and dragonfly, both of them are biocontrol agents because ladybird is going to kill the aphids and your dragonfly is going to kill the mosquitoes. Okay. Now, next thing, I will talk about trichoderma first. So, trichoderma is a fungus. It's a free-living fungus which is there found in the roots of the plant. Now, in the roots only there are other very bad funguses also. So, trichoderma kinds of feeds on these bad funguses. So, free-living fungus used to control several plant pathogens. So, this trichoderma, and remember there is trichoderma polysporum who releases cyclosporin A. Please remember that. There is trichoderma here, there is trichoderma there also. Now, you have Bacillus thuringiensis, a very famous thing. So, Bacillus thuringiensis is a bacteria, my dear children, and what does it do? What does this particular bacteria do? Basically, it has a gene in its body which produces, so the gene's name is cry gene, and it produces what? A protoxin. A protoxin which is in the inactive state. When does it get active? When the pest which is there, it takes in this bacteria. At this point of time, this toxin which is there, it gets activated. You get the active toxin, and this toxin is going to damage the epithelial cells of the mid-gut region, my dear children, of the pathogen, and the pathogen is gonna die. That's how it works. Always remember, mid-gut of the pathogen that is going to be affected, and the epithelial cells are the one that are affected. So that is the role of Bacillus thuringiensis. You can basically take the dried spores, put it in water, mix it, and then apply on the plant. Okay, or then you can do one more thing, that is you can take this gene, you can do recombinant DNA technology where you take this gene of cry gene and put it in the plant itself. So plant itself will produce protoxins. That is also done. Now, baculoviruses are pathogens that attack insects. They are viruses, my dear children. They attack insects and other arthropods. The majority of baculoviruses used as biological control agents are in the genus Nucleopolyhedrovirus. Please remember the genus name. Okay, they are in the genus Nucleopolyhedroviruses. Now, there is one very important thing that is about biofertilizers. What do you mean by that? Biofertilizers again are organisms. Again, fertilizers, if I put no, there are chemicals which will really lead to what? A change in the fertility of the soil and things like that. I don't want to hamper any of it. So, basically what do? I use biofertilizers. So, these are organisms which increases the fertility of the soil. They add up certain nutrients to the soil. So, what do we have over here? We have the first biofertilizer, which is a bacteria, Rhizobium. Okay. We have three bacteria over here, Rhizobium, Azotobacter and Azospirillum, my dear children. It forms, the Rhizobium, it forms symbiotic association with leguminous plants such as pea plant and things like that, and they fix nitrogen. You know, in atmosphere, nitrogen is in the form of N2. So it is like this, which has triple bond. Which has what? Triple bond. It's very difficult for the plant to break it. So obviously there should be some bacteria which could be breaking it down and provide the plant nitrogen in the form of nitrates or nitrites. So Rhizobium does that. It forms symbiotic association with the leguminous plants and fixes nitrogen. Apart from that, you have Azotobacter. It's a free living nitrogen fixing bacteria in the non-leguminous soils. Then you have Azospirillum. It is associative nitrogen fixing bacteria. You don't have to remember at least this, at least remember that Rhizobium, Azotobacter, Azospirillum, all of them are going to fix nitrogen and they are examples of bacteria. Now, in bacteria, you have different group, that is cyanobacteria. You know, blue-green algae. they have the ability to do photosynthesis. They have the ability to do photosynthesis as well, my dear children. So they are Anabaena, Nostoc, Oscillatoria. Okay, so they fix nitrogen. Apart from that, they also produce organic matter. So they fix nitrogen, they produce organic matter because they are responsible for photosynthesis. They fix atmosphere in the paddy fields, they are used very much, and other aquatic environments also they are found very much. Remember always this question remember always that this question also can come, that is, give examples of cyanobacteria which acts as biofertilizers, give example of plain bacteria that acts as biofertilizers. So you should know this difference, very very important. Now if fungus also, you have biofertilizer which is mycorrhiza. Mycorrhiza is basically a combination of plant and the fungus. So plants do photosynthesis, they provide the fungus with organic matter. Okay, and what does the fungus do? Fungus basically seep into the soil and take in phosphorus. Remember, it is phosphorus, not nitrogen. So phosphorus taken and given to the plant. Okay, and this fungus which forms a symbiotic combination, that is mycorrhiza, is from the genus Glomus. This is again a very important question. Glomus. So it forms symbiotic association with plant roots, enhancing phosphorus absorption and improves what? Plant growth as well. So Glomus, remember that. Okay, these things, remember, important thing. Baculoviruses from again the genus Nucleopolyhedroviruses. These are the small things that you might forget. Now, describe the role of microbes in sewage treatment. Draw a neat labeled diagram of typical biogas plant. Can you do that? So this is the neat labeled diagram of a typical biogas plant. So over here, my dear children, this is the place where you put the dung and the water. Why dung? Because the cow dung has, in the rumen of the cattle, in the part of their intestine, my dear children, this is rumen. And in the rumen of the cattle, you have, in the part of their digestive system, they have rumen, and in that you have methanogens or methanobacteria. They produce methane. Okay, so basically it's not just for methane that they are there, they help in the breakdown of cellulose indirectly, not directly. Plants are being taken up by the cows and all, and it is very difficult to break down the plants because they have a cell wall made up of cellulose. So there are methanobacterium which helps in the breakdown, not directly but indirectly. So they are there in the rumen of the cattle. In us it is not there, which is why we cannot eat raw plant and all. We have to boil it so that we can break the cell wall. So basically in the cow dung also this methanobacterium will be there. I'll mix it thoroughly with water and then let it go over here in the digester. This digester is in anaerobic condition. There is no oxygen up over here. If there is no oxygen, my dear children, what will happen? There will be anaerobic respiration happening over here. It will break down and will give rise to, the organic matter is being broken down and gives rise to what? The biogas that contains of, tell me, it is methane, it is H2S and also carbon dioxide, all this thing. So this biogas is being taken, and then whatever is left behind is put over here. Okay, from here it can be used as fertilizers or manure as well. Cool. Describe the role of microbes in sewage treatment. We already spoke, so please talk about aerobic bacteria, then anaerobic bacteria, and that is methanogens and all these things. Okay. Now, explain the role of 3 organisms as biocontrol agents. Will you remember? Can you remember 3 organisms are biocontrol agents? Ladybird, dragonfly, and then you had baculoviruses, then you had trichoderma, this is fungus. You have BT, Bacillus thuringiensis, all of them. What are phlox? Phlox, as I told you, is a mesh-like structure which is formed of aerobic bacteria as well as fungal filaments as well as the extracellular. Outside the cell, they are releasing something, that is extracellular content that is being formed. Okay, it acts on the organic matter. Now, what is the significance of BOD? BOD represents what? Biochemical oxygen deficiency, right? Yeah, biochemical oxygen deficiency, which basically suggests how much amount of oxygen is required by the aerobic bacteria to break down the organic matter in 1 liter of water. So if it is more, that means the pollution is more, there is too much amount of organic matter. If it is less, then the pollution is less. Now give an example for a fungus found in mycorrhiza. It is Glomus. It is what? Belonging to the genus Glomus. That's all. Have I written everything? Yes. You can just go through it. More pollution, poor water quality. Phlox are masses of bacteria and fungal filaments in sewage treatment. They have been breaking down organic matter in the wastewater. Okay. Name the bioactive molecule for the following. Bioactive molecule, they active, very active. These are active molecule, my dear children. So we have a clot buster agent. I told you who is the one that busts the clot. It is streptokinase. And streptokinase is released by what? Let me increase my energy. I think you also will be done. Okay, Jasmine, you can do it. Okay, streptokinase is released by what? Streptococcus, which is what? A bacteria, my dear children. Okay, so this is released by a bacteria. Now, immunosuppressive agent, that is cyclosporin A. It is produced by a fungus, that is Trichoderma polysporum. Okay, going ahead, a blood cholesterol lowering agent which is there in our body, it is what? Statins, and it is produced from Monascus purpureus. Monascus, M should be capital, okay? Monascus purpureus, which is again something, fungus, I think, yeah. Now, going ahead, what are biofertilizers? We already gave the example. Can you give an example of biofertilizer? Azotobacter. Remember something? Oh my god, azospirillum, rhizobium, all this thing, rhizobium, azotobacter, azospirillum, anobanum, nostoc, Nostochluria. So when you're asked about bacteria, you can just write this, and if you can just write cyanobacteria is equals to all this thing. Okay, have a separate category for them. Okay, just a second. I think we are up. We're finishing this. Okay, with the help of a clue in the given brackets, complete the passage with appropriate answer. Okay, I'll tell you first. I'll tell you the story first, and then we'll go on. Okay, so do you know Alexander Fleming? So there was a guy whose name was Alexander Fleming, and he was rigorously working on something. Okay, that is Staphylococci as a bacteria. He was working on that, and then one day he didn't wash the dishes where he was culturing this particular bacteria. So it was kept like that. It was kept like that. The other day he came like, oh my god, I put it like that. It would be very dirty by now. The bacteria would have grown like this. But then he comes to a surprise to understand that, oh my god, something happened. The bacteria is not growing. The bacteria is not growing over here. It's just there. So what is that thing that would have led to stopping of the bacterial growth? He went ahead and he searched that, and he found that there were certain fungus. What was this fungus? It was Penicillium notatum. It was Penicillium notatum. This fungus was there. And probably when he went ahead and searched that, he got to understand whenever this particular mold is there, Penicillium notatum's mold is there, this bacteria was not growing. He got to understand that probably something is released over here which kind of scares off the bacteria. So what is that thing that scares off the bacteria? It should be an antibiotic. It should be an antibiotic. So this Penicillium notatum is releasing an antibiotic whose famous name is what? Penicillin. I might be wrong with the spelling. Please check the spelling, okay? It is what? Penicillin. So later, its actual use and everything was put down by some Ernst Chain, Harvard Flore and people like that. But Alexander Fleming was the person who introduced it. Later it was, and he discovered, so we say he discovered the penicillium, antibiotic, that is penicillium. Later it was used, okay, used for good purposes by Ernst Chain and Harvard Flore, who went ahead and did so many things. So it was used in World War II also for treating the American soldiers which were there, and they actually got Nobel Prize for it. How many people? 3 of them. They all got Nobel Prize for this. Now we can solve this. So with the help of the brackets, okay, while working on dash, what? Staphylococci, okay. Scientists observed the growth of a mold, that is Penicillium notatum, okay, which does not allow the bacteria to grow. He found out that it was due to a chemical penicillin produced by the mold. This antibiotic was used to treat soldiers of what country? America, wounded in World War II. Dash, dash and dash scientists were awarded what? Dash, dash scientists as in Alexander Fleming, Ernst Chain and Harvard Flore were awarded Nobel Prize in the year dash of this discovery. I forgot the year, 1945, yes, 1945 for this discovery. Works. They had fun learning that, yeah. Now going ahead, assign the role of following microbes. Do not see it, okay. So lactobacillus in milk is therefore converting milk to curd. Rhizobium in the nodules of legumes, my dear children, it fixes nitrogen. Trichoderma in root ecosystem kills other fungus. It is acting as a biocontrol agent. Methanogens in the rumen of cattle, it basically helps in the breakdown of cellulose indirectly. Okay. Glomus in mycorrhiza helps in absorption of phosphorus. It increases the nutrient absorption. So let's see. Lactobacillus converts lactose into lactic acid, helps in curd formation. Rhizobium in the nodules of legumes fixes atmospheric nitrogen into ammonia, aiding plant growth. Trichoderma in root ecosystem acts as a biocontrol agent against soil-borne plant pathogens. Methanogens in the rumen of cattle helps in methane production by digesting cellulose in the stomach. And glomus in mycorrhiza enhances the nutrient absorption, especially phosphorus in plants. Works? Done? Deal? Are we all cool? Yes. And we have completed that also, my dear children. Please go through everything that I have told you as of now, otherwise you will not have any understanding of things. And the next chapter is Biotechnology Principles and Processes. We'll come again to it, take a break, this time take a 5-minute break and come back. Okay? And do let me know if you have any doubts in the comment section, and do let me know if you are enjoying this session. Is this session useful or not? This also gives me a little bit of motivation, right? Yes. So let's get ahead with biotechnology principles and processes, starting with the first question. Name 2 bacteria that were used for developing recombinant DNA by Stanley Cohen and Herbert Boyer. So this was the first recombinant DNA that we could create. So what were the name of 2 bacteria which were involved? It is Escherichia coli, that is E. coli, and also Salmonella typhimurium, that is going to cause what? Typhoid. Now my dear children, how was it used? You should be having a background idea of this as well. So what was being done is that Salmonella typhimurium, if you talk about that, it has its own DNA and all the things. Apart from that, in all the prokaryotes, they have something that is known as plasmid. What do you call it? Plasmid. So plasmid is the extra chromosomal DNA. It is what? Extra chromosomal DNA, which is just there. It does not code for proteins, nothing. It's just there. It has its own origin of replication. It keeps on replicating as well. Now, the thing is, this particular plasmid was taken out. Okay. And inside this particular plasmid, what do you call it? Stanley Cohen and Herbert Boyer, they basically inserted an antibiotic resistant genes. An antibiotic resistant gene. What do you mean by that? Basically, that means that whoever have got this particular plasmid, they would be resistant to certain antibiotic. Okay, certain antibiotic. So this was being put up over here. And then, if you insert the plasmid back into Salmonella typhimurium, what will happen? This particular guy will be having that resistance, will be having that power to resist the, against an antibiotic, and basically it won't die. We want it to die, right? We want it to die, but we just wanted to test it whether basically we can create an antibiotic resistant, recombinant DNA or not. We just wanted to check that. So what did we do is that we put this particular plasmid into the next bacteria, that is E. coli. We put this plasmid there, and then we try to basically grow this particular E. coli in a medium that was containing the antibiotic. In normal case, the E. coli should not have survived, but since it has the plasmid that contains the antibiotic resistant gene, what will happen, my dear children? It will survive, and that's how we got the first recombinant DNA. Okay, just wanted to check it out whether recombination is possible or not, and that's how they found it. So this is the story. Please remember, Escherichia coli and Salmonella typhimurium, this is the one that causes typhoid. Going ahead with the next question, differentiate between exonuclease and endonuclease. Both of them are restriction enzymes, my dear children. Restriction enzymes, you know, they are the ones, they are known as molecular scissors, because they are the ones that cut the DNA. When do you need cutting? I just told you that over here, in this plasmid, the antibiotic resistant gene was being put up. So don't you think there is a need to cut the DNA segment which is already there? Yes. And also you have to cut this antibiotic resistant gene from somewhere else and put it over here. So you need molecular scissors that are restriction enzymes, and you have 2 types of them. One is exonuclease, the other one is endonuclease. As the name suggests, endonuclease means it is going to cut within, inside, okay, endo, it will enter and it will cut. Exonuclease, it will be cutting from the ends. So it removes nucleotides from the ends of the DNA strand, whereas this cuts DNA at specific sites within the strand, my dear children. Now mode of action, cleaves the nucleotides one by one from the terminal ends. From the terminal ends, it is going to remove the nucleotides one by one. But over here, it breaks the phosphodiester bond at the internal sites. At the internal sites, you know, one nucleotide is attached to another nucleotide with a phosphodiester bond. It is going to cut that, and that's how it basically breaks down the DNA over there. Now example is exonuclease 3. Over here we have EcoR1, we have HIND3 and everything. Okay. So HIND3, EcoR1 is there, exonuclease 3 is over here. Now this is not given in your NCERT. If you want, you can just go through it for your understanding. Okay. Now EcoR1 and HIND2 is the first. If I tell you, HIND2 was the first enzyme, okay, restriction enzyme to be found out. HIND2, and I'll tell you one more thing. How many, okay, so basically these enzymes cut at sites. They have specific sites for cutting. They do not just randomly cut here and there. They are very specific with the action. They have specific site. You call them recognition sites. What do you call it? You call it recognition sites, my dear children. So the recognition sites for HIND2 has 6 base pairs. Has what? 6 bases. 6 bases. Similarly for EcoR1 also it has 6 bases. What are the 6 bases? GAA, TTC. In 5' to 3' direction. GAA, TC. That is how you can remember it. I sing very badly, but I hope you remember it. So GAA, TTC is the thing. So when someone sang, that is me sang, I got TC because that it was that bad. So you can remember like that, TTC. Okay, so double TC. So anyways, 5' to 3', that is the restriction site for EcoR1, my dear children. And in this restriction site, it cuts between G and A. It makes a cut between G and A. Okay, with this I remember there is a question on sticky ends. On sticky ends, what is the use of sticky end? How is sticky end formed? I have not included that question over here. Please go read through that question. It is there in 5 marker or 5 marker, I think. Yes, in the 5 marker video. Please watch it. Very important, very, very important. Okay. Children, please understand why I have not included everything over here. If I go ahead with PYQ and question banks, do you think how long will this video be? Will you be able to hear me for this particular very long time? No. Even if some people love me, still they cannot hear. They cannot hear. I know, my dear children, even I love you, even I cannot talk for that long. You get it? So it's totally understandable. So yes, I'll go ahead. Okay, so please EcoR1 and HIND3 and what else I had to say. One more thing I had to say. Okay, when restriction enzymes were we found out, okay, what was the thing? So basically it was found that in some prokaryotes, in prokaryotes, they were having 2 enzymes. One enzyme is basically adding methyl group on the genetic material. One enzyme is adding methyl group on the genetic material, another enzyme is cutting the genetic material. So people didn't understand. Then they got to understand that basically when some infection happened, for example, any random organism put its DNA inside the body of a prokaryote. Then this prokaryote obviously has to cleave this. Otherwise, what will happen? It will slowly integrate into its own genetic material. So to cleave it, they had restriction enzymes, my dear children. And if they have restriction enzyme, these restriction enzyme can act on them also, right? On their own genetic material. So to safeguard, they added methyl group. So safeguard, they added methyl group. So whenever there is a methyl tag, it was not being cut. It was not being cut. So that was actually a way to safeguard themselves. This is a way to safeguard themselves. But now we are using this restriction enzyme for our own purposes. I hope you got it. Now going ahead. Explain the convention for naming restriction endonuclease scientifically. Okay, let's see. So EcoR1 is a thing. EcoR1. So R1 you write in Roman letters, not like 1. Okay, don't do that. Now what does the first letter? Uppercase. E stands for Escherichia, which is the genus. From the Escherichia coli, it represents what? The genus of the bacterium. Now the next 2 letters, that is lowercase, in lowercase, okay? It is representing the species of the bacterium, that is CO for coli, that is a species name. Now we have additional letter, if any, that is for example strain. This represents the strain. R represents what? The strain. So it represents the strain, that is RY13 of Escherichia coli. RY13 of Escherichia coli. Now the Roman numerical, it indicates the order of discovery. For it was the first one to be discovered from that particular strain, so I put 1. Okay, will you be able to answer like this? Please answer like this. Okay, going ahead. Name the technique involved in separation and isolation of DNA fragment. Which dye is used to stain gel to make the DNA visible under UV light? So what is the technique for separation of DNA? Tell me, what is the technique that is used for separation of DNA, my dear children? For separation of DNA, we have gel electrophoresis. How does gel electrophoresis chamber look? I'll show you. I have a picture ahead, but see over here itself. Okay, so we have a cathode towards this end and an anode towards this end. Now, if you do not know, I'll tell you, anode is positive, cathode is negative. Anions are negative, cations are positive. Okay, cat has pause, so I remember like that. Cations are positive, but cathode is negative and anode is positive over here, ulta. Okay, now there are certain wells over here. In this wells, we are going to put the DNA. What will happen? Depending on the fragment size, they are going to separate. So the shorter one will be able to move fast. Why is it moving towards the anode, ma'am? Because, my dear children, DNA which is there, it is negatively charged. So negative repels. So it will move away from it and go towards the anode under an electric field. What is this gel that is used, ma'am? It is known as agarose gel, and you get it from the seaweed. This is what? Agarose gel, my dear children. Ma'am, do they move on the top? No, my dear children. Basically, the DNA moves with a sieving effect. That means it moves within it. My dear children. Okay. Now, the smaller ones move ahead and the larger ones will be coming slowly. Now, if you want to see the DNA fragments, you cannot see it like that. So, basically, you put a stain which is known as ethidium bromide. Very much important. You put ethidium bromide. After putting the stain, you will be like, ma'am, you were making fun. No, it's still not visible. So you need UV radiation also. You need UV rays also. So when we put UV rays, what happens? Wherever the DNA has taken up the ethidium bromide, it will be giving you a bright orange color. It will give you a bright orange color. Please remember the name of the color also. It is what? Bright orange color that will be visible. Okay. Now, finally, if you want to remove this DNA, what do you call this? Removal or taking down of this DNA, separation of this DNA from here. What do you call this? This process is known as elution. This is known as elution. It starts with E, E-L-U-T-I-O-N. Okay, so I hope I answered everything. Technique for separation and isolation of DNA fragments is basically gel electrophoresis. At the end, basically, when you isolate the DNA from the gel electrophoresis chamber, it is known as elution. Basically, when you cleave it out. Okay. Now, dye used for staining DNA in gel electrophoresis is ethidium bromide, that is ETBR, and ethidium bromide binds to DNA and fluoresces under what? UV rays, UV light, making the DNA bands what? Visible. Okay, not UV rays, UV light, and it gives bright orange color. Works? Sure. Next thing, and draw a neatly labeled diagram of typical agarose well. Now you will be able to make it, no? So these are the wells that we have. This is the cathode. Please mention the cathode towards this side and anode towards that side, and show the separation. The smallest bands are here, the largest bands are over here, because they move really slow. Next question, draw a neatly labeled diagram of PBR322. This is the most important diagram from this particular chapter. Okay, please remember you have to draw this. How do you draw this? Listen. So this is one anda, second anda. Over here, make a line like this, make a line like this on opposite side. Okay, this is tetracycline resistant gene. This is tetracycline resistant gene. Tetracycline resistant gene as in this particular plasmid, if it's there, this particular organism that has a plasmid would be resistant to the action of tetracycline. Tetracycline is basically an antibiotic. So it is tetracycline resistant gene. Over here you have another antibiotic, that is ampicillin resistant gene. This is what? Ampicillin resistant gene. I should have not written over here. So I'll just write it over a U. Ampicillin. I'm just writing it like this, okay? You understand, I suppose. Similarly, I'll just erase this and I write there. Tetra. Okay, fine. Now, opposite we did. Now, here you have to make two lines. Similarly. Okay, so this is the ORI site, origin of replication. This is where the DNA polymerase will come and attach and do the whole replication process. Now, this is an ROP site. ROP is a very important, ROP site is responsible for replication itself. It is responsible for formation of protein that is responsible for replication. So it is ROP site, my dear children. Okay, now listen to me. The restriction enzymes also need to have some sites. Okay, they will also be cutting at specific sites. So where are these sites located? I am going to talk about that. So in each you have two. So over here there is one that is known as BAM H1. Okay, now here you have one more, that is SAL1. BAM and SAL, BAM and SAL, BAM, SAL, BAM, SAL. So it's BAM H1 and it is SAL1. Now over here you have ampicillin resistant. Here also you have two. One is PVU1 and the other one is PST1, PVU1 and PST1. Now PVU1 says, I have my friend PVU2 also. Where is it? It is over here. It is over here, my dear children. So there is a restriction site over here, and this is known as PVU2. This is known as PVU2. So first make this ampicillin tetracycline. Done. Now you have to make two each, BAMH1 and SAL1. Here PVU1 and PST1. Now over here in ROP site you have PVU2. Now here you have three of them. One is the very famous EcoR1. Okay, I have to look. Yes. One is yes. We have EcoR1, and then we have CLAV1. So EcoR1, CLAV1, and then a very important thing that we have is HIND3. We have HIND3, my dear children. Okay, so EcoR1, CLAV1 and HIND3 is also there, and with that we are done with PBR322. This is a very important plasmid. 322. So yes. Okay. How you can remember this? 3, 1, 2, 3. 2 each, and over here 2. So 2 over here, 2 over here, 3 over here. So 322. I made it now. Okay, so works. Chalo, let's get ahead. Draw a neatly labeled diagram of stirred tank bioreactor. Again, a very important diagram. Okay, so over here, please make the whole thing. Let an entry for the sterile air which is there. Over here, you have what impeller for basically mixing out everything which is there. The air should be mixed, the culture broth should be mixed. Okay, and acid base for pH control. You have a thing like that. For steam for sterilization, you have another outlet and inlet. You have foam breaker. You have motor. Basically, the motor only it can rotate. So this is the whole setup of a stirred tank bioreactor. Now we have sparged tank bioreactor. Until now it has not been asked, but we never know when it comes. So basically sparged tank is basically you're taking air bubbles and putting it inside. In that way it is a lot more better. If you're asked which is better, it will be sparged tank which is more better. Sparged, oh my god, sparged stirred tank bioreactor. So this is sparged one. So over here, as I said, the air are in the form of bubbles, and over here you have an impeller which moves. There is gas entrainment, and there is increased surface area for oxygen transfer. As you can see, bubbles are coming, and yeah, that's all. Okay, there is nothing much to explain over here. It can be drawn as such. I think you have to by heart over here. Okay, chalo. Next, what is downstream processing? Write a note on downstream processing. Listen to me carefully. So basically. When you do the whole process of recombinant DNA technology, at the end you get your product. Every gene which is there, it is going to code for a protein. Okay, for example, I put a gene in E. coli and it made the protein. Now I have to separate this protein from everything else. I have to make sure that's pure, it's clean, everything is good. This is known as downstream processing. It refers to the series of processes required to purify and extract the desired product from the culture after completion of biosynthetic phase in biotechnological process. After all the phases, when I remove it, when I'm taking it out for purification, okay, after purification and extraction, this is known as all the processes which is involved in purification and extraction is known as downstream processing. The first one and the major one which is involved over here is separation and purification. You're separating things from all the culture medium and everything, and then you are purifying it. This is the actual one. This is the actual one. And after this comes a lot of other things. So in general, in your book it is written downstream processing involves separation and purification. So make sure you're writing this, and then you go ahead with anything else. So everything else comes like the next part after downstream processing. So you have formulation, where the purified product is converted into a usable form, that is liquid or powder, whatever is there. Okay, you preserve it as well, you do preserving as well. Then quality control and testing is there, where the final product is tested for purity, safety and effectiveness. Then you're packing it and you're storing it. The product is packed and stored under appropriate condition. Significance: it is crucial step in industrial biotechnology, ensuring that the final biotechnological product like antibiotics, vaccine or enzymes is safe, pure and it is market ready. So separation and purification first, then you preserve it and you formulate. There is formulation, there is quality control and testing, there is packaging and storage as well. Okay, going ahead, write any 4 tools used in recombinant DNA technology. Okay, first tool is restriction enzyme. You know, it's for cutting. Now listen, you cut. Now suppose you cut a plasmid over here and you are joining something new. Will it join like that? automatically? No. You need an enzyme which is known as DNA ligase, which is ligating things. So DNA ligase is therefore ligating this thing. Then now obviously this particular guy will be put in some organism, okay, or basically this particular thing which is used, which is carrying, okay, this particular thing which is used, which is carrying the important gene. What do I call it? It is carrying the important gene. What do I call it? I call it as a vector. A vector. So such as plasmids and bacteriophage, which is used to transfer the DNA, it is going to act like a vector. Okay. Now next thing, host organisms. Suppose this particular thing, now we are going to put in E. coli, because E. coli is the one thing where the production will be happening now. What do you call this E. coli as? You will call this E. coli as host organism where the actual production takes place. So what are the tools that I need? I need restriction enzyme, I need DNA ligase, I need the vectors who can carry things, I need the host organism with at the end which will be used for cloning and expression for the actual product. Okay. Now methods. Mention any two methods of introducing alien DNA into host cell. Into host cell, how can we put the alien DNA? You have plasmids. You have plasmids, my dear children. But apart from that, if you're asked about methods, okay, I'll say let's put down two methods, microinjection and biolistic, which is known as gene gun. What do you mean by microinjection? For example, there's an animal cell. There's an animal cell. I do not want any vector to carry it. I do not want any vector to carry it. I would just directly want to make it I want to make a change so that it has my DNA. So I use microinjection technique where with the help of a micropipette, okay, basically I am going to directly inject the DNA inside the nucleus. This is known as microinjection. Please remember, this happens only in animal cells. This happens only in animal cells, and this is an example of vectorless method of transmission. You do not need any vector over here, you're just using this, that's all. Okay, no plasmid, nothing. Now going ahead, biolistics. Now suppose the same thing is a plant cell. Plant cell has an additional layer which is known as cell wall. I cannot use a micropipette to basically make my DNA enter. So because it is a very thick layer that I have over here. So I'll use a gene gun. I will use what? A gene gun. This is known as biolistic method or gene gun method, where basically I have microparticles of gold and tungsten. I have microparticles of gold and tungsten, and I'm going to cover it with the DNA that I want, not like wrapping the DNA. Okay, there is a technique involved in it. So the DNA will be all around the microparticle which is there, and finally it is bombarded with high velocity such that it lands up in the nucleus. This is seen in plants. Again, do you see any vectors involved, any living vectors involved? No, my dear children. So this also acts like a vectorless technique. This also acts like a vectorless technique. There is one more, that is electroporation. There is one more, that is electroporation, where you are having you're passing electricity and you're making pores, and a lot of things are there. So please, I request you to watch that 5-marker, 2-marker video where I have told you it won't take too much of your time. If you go chapter wise, it is not going to take too much of your time, and you will feel that you are doing a wholesome preparation. You get it? So there are other methods, electroporation and things like that, that is used for bacteria. Now these are the vectorless methods used for animal and plants. Now there is for bacteria as well. There is electroporation that I was talking about. Okay, where you use cesium chloride and all these things. Cool. Now we have name the stains used to visualize DNA fragments. I told you already, it will be what? Ethidium bromide, my dear children. It is ethidium bromide. Going ahead, briefly explain the steps involved in recombinant DNA technology. So first, what do? You do isolation of the DNA. You do isolation of the DNA. As in, for example, I am the organism. In my cell, there is RNA, there is protein, there is DNA. I have to exclude the RNA, exclude the protein, and I just have to take the DNA. So what do? I treat my cell with RNAs. I treat the cell with RNAs, my dear children. This is going to be really important for you to know. So this is RNAs which is going to cleave down RNA. I won't have RNA then. Then what about proteins? Proteins are cleaved down with the help of protease. Proteins are cleaved down with the help of proteins, so all proteins are removed. Now the lipase which is there, lipase is going to act on lipid like fats and everything. It is going to break down that as well. Now it is about an animal cell, that is my cell. What about a plant cell? For a plant cell, instead of getting into the DNA, instead of getting into the nucleus, first thing that you have to do is you have to get inside the cell. But the cell has cell wall. What do? So cellulose is something that makes up the cell wall of the plant cell. So what do? You treat it with cellulase. You treat it with cellulase. Very important thing. Again, I have explained it there. Okay, cellulase is for plant cell. Now, if it is a fungal cell, then what do? Again, there is a cell wall, and this cell wall is made up of chitin. So you need chitinase. Now, what if it's a bacteria? What if it's a bacteria? Bacterial cell wall is made up of peptidoglycan. So what do you need? You need at least lysozyme, the one that is there in our tears. Okay, so lysozyme is taken, and that's how you break it down for bacteria. You get it? Did you get it? And once you're getting inside for removing proteins and RNA, for proteins you have protease, for removing RNA you have RNase, for removing lipids you have lipase and things like that. At the end you get DNA. The DNA does not separate out itself. You have to add chilled ethanol. If you add chilled ethanol, my dear children, that's when the DNA will be separated. Please remember that. Okay, now isolation of DNA is done. Now second thing is cutting of DNA. You have to cut the DNA that you want, and you have to cut the DNA where you want to enter things, right? So what do you use for cutting? You use restriction enzymes, that is endonucleases, to cut at specific sites. Example you can give like EcoR1 and things like that. Tell that it cuts in 5GATTC site. 3 also 5GAATTC 3 dash site. Now it is going to cut between G and A. Mention that. Now after cutting, what do you want to do? You don't want big fragments, you want smaller fragments. So you have to separate them. For that, what do you have, children? You have gel electrophoresis. So you make them run through gel electrophoresis chamber, and then it is visualized with the help of a stain and also under UV light. Then what do you do? Basically you're taking the DNA, you're going to put the DNA into the vector. That is probably a plasmid or whatever is there. So you have to ligate it to a vector with the help of what? DNA ligase. With the help of DNA ligase. Now here comes the role of sticky ends. You can talk about sticky ends as well. Now after that, you're going to put this vector into what? The host organism, which is going to basically in their body only this particular plasmid is going to replicate, it is going to produce the thing. So it is going to be put inside the host cell. Example, you have E. coli. After that, once you have put in the host cell, you have to understand which cell has which plasmid. The recombinant plasmid that we have made, my dear children, which has the new gene and all, is it there in the E. coli or not? Is it there in the E. coli or not? So for that, we have to select the transformed cell, my dear children. We have to use this, we have to select the transformed cell. Transformed cell as in the one that has taken up the plasmid. The one that has taken up the plasmid, my dear children. Okay. Now there is two E. coli. There is two E. coli, both of them, one of them have taken up the plasmid, one of them has taken up the recombinant plasmid. Okay, recombinant plasmid. So how do we understand it, which of them has taken what? So basically, either way you will call them transformed. Either way you will call them transformed because both of them have taken up plasmid. We do not know which one has taken which plasmid. Okay, so either of them will be known as transformed cells, but not recombinants. Only one is recombinant, the other one is not. Remember that. So anyway, how do you find this transformed recombinant? We have selectable markers for that, my dear children. We have selectable markers. So I told you the plasmid that we have taken, suppose. Okay, the E. coli plasmid that we have taken over here, it didn't have antibiotic resistant gene, but now we are putting an antibiotic resistant gene over here. Or the vector that we have put in, okay, wait. This is an E. coli, and the vector that is there, it has an antibiotic resistant gene, my dear children, and is the one that we have put. So this antibiotic resistant gene, we are putting it there because it is going to be acting like a marker. And here we have our gene of interest as well. Now there is another one, there is another E. coli which is basically taken up a plasmid, but not the one that has the recombinant gene. So how do you identify? We treat it with with the same antibiotic. If it is able to survive, that means it has taken up the one that we are looking for. Both of them are transformants. Both of them are transformants. Both of them have taken up plasmid. Okay, they originally didn't have this particular plasmid, but this is the one that has a gene of interest as well. We will get to know when we have put the antibiotic, if it is able to survive in that. Okay, there are different methods. We will be talking about that. So please do not worry. Okay, now I'll be talking about that. So selection of transformed cell, and then also selection of transformed and recombinant cells. So transformed as in the one that has taken up the plasmid, any new plasmid, and recombinant as in one has taken up the recombinant, the new combination of plasmid. Okay, then after that, expression of the gene. Yes, the genes will express, it will form the protein, whatever is needed, and then there will be downstream processing. These are the steps that are involved in recombinant DNA technology. Got it? Going ahead, we have what is polymerase chain reaction. Before that, I'll see if I have a question. Yeah. So with this I can explain you a lot of things. Okay, this the transform and recombinants and things like that. What do you mean by insertional inactivation? Explain with respect to beta carotesidase enzyme. How are recombinants and non-recombinants identified? So listen to me carefully. Okay, so you have a plasmid, and this plasmid has a site which is LAC-Z gene site. Okay. Now LAC-Z codes for beta galactosidase. Beta galactosidase. Now whenever beta galactosidase is there, this enzyme is there, and whenever there is a coloring agent being put and a coloring substrate being put, for example X-gal is substance like that, what will happen is that this beta galactosidase will act on it and hence will give a blue coloration, This is a general scenario. So whichever organism has taken up this plasmid will be giving a blue color colony. Okay. Now what do I do is that I bring out certain changes over here. I remove this LAC-Z gene, and put my gene of interest over here. This is my gene of interest. Will there be production of beta galactosidase? No. So this is an example of insertional inactivation. You are inserting something which is why other thing is getting inactivated. Now there is no gene of interest. There is no LAC-Z. There is gene of interest. Okay, there is no LAC-Z, but there is gene of interest. Does this gene of interest go make any changes in the coloring agent? No. So the vector that has, so the host organism that has this now, my dear children, suppose this is the one that has it. Okay, and this is the one that had the normal one, that had the normal X-gal one. So I am marking it in green. So what will happen is that when we are adding the coloring agent, we will see that few of them will be giving the blue color, and few of them they are not going to give any color. It will be white color colony. In this way, I will be able to identify that the one that is having white color colonies are the one that has taken up the plasmid with what? The gene of interest. So this is the recombinant plasmid that we have. So white color things, we are able to identify that it has it, and the blue color ones basically they do not have. So they have taken up the plasmid, but not the plasmid with the recombinant ones. They have taken up the plasmid, but not the plasmid with the recombinant ones. So what will happen? Both of them are transformed and both of them have got the plasmid, but one has the recombinant one, another one does not have. How did we identify? Because this one did not show insertional inactivation, it's still showing what? Blue color. But this one, my dear children, it is showing white color. That means it has the recombinant one. Get it? This is known as blue-white colony separation. This is how you find the recombinants and the other one. Okay? The answer is written over here. Insertional inactivation, LAC-Z gene, in LAC-Z gene, it overcomes the need for dual plating for antibiotic plates. Okay, there is no need for plating it separately. There is no need of two plates. Okay, there is also a question. Okay, do we need two plates over here? No. In one plate only, we put the coloring substrate and we find that some of them will give white color colonies, some of them will give blue color colonies. Okay, there is no need for dual plating. This is a very important thing. I will tell you about one more thing. Okay, I will talk about the PBR322 plasmid. This is a different plasmid. That is a different plasmid. In that, we need two platings to be done to identify which one is a recombinant one. Please understand. Once more, I will tell you. So there is a plasmid that no one of them had. This is the plasmid that none of them had. This had the LAC-Z gene, and in this one we had removed it. Which one is the recombinant plasmid? This is the recombinant plasmid. This is the recombinant plasmid. Suppose both of them was there and the E. coli was there. We are going to put them, and inside that we found that both of them basically they took up the plasmids. They are taken by both this E. coli has taken up the plasmids. So we say that both of them are transformed. They have the plasmid, that means they are transformed. We do not know which one is the recombinant. To know that, we put the coloring agent, and then we found that basically here probably the gene might be producing beta carotesidase, and hence it gives blue color colony. That means it is not the recombinant. Here they saw that it was not changing the color. That means it is giving a white color colony. That means it has the recombinant plasmid. Got it? So you just need one plate. In that one plate you will be able to identify it itself. So, foreign DNA inserted into beta-carotesidase gene is inactivated, chromogenic substrate is added, non-recombinant, that is no insert, will produce blue colonies, and the recombinants produce what? White colonies. So, white colonies is equals to recombinant DNA. I hope you got this. Now, going ahead with this question that we have over here, what is polymerase chain reaction? Name the bacteria from which the polymerase enzyme is used in this technique. No. Write the schematic representation of this technique. So, over here, this is polymerase chain reaction. Polymerase chain reaction is one very important thing which helps in the amplification of even little amount of DNA. It has done in these many steps. So, what do you have? You have denaturation, my dear children, where what do you do? Suppose you have a double-stranded DNA. You're going to separate it. This process is known as denaturation. After separation, what you're going to do? You're going to do annealing. Annealing means there are certain primers. There are certain primers which are very much, what is the use of all this process? We want to make copies of DNA, and for making copy of DNA, what do we need? Which enzyme do we need? We need DNA polymerase, and DNA polymerase cannot start off a new branch like that. It needs primers. So, the process of attachment of primers is known as what? Annealing. Okay, and after that, when you provide DNA polymerase and also the nucleotides which are needed, what will happen? The next process will happen, that is known as extension. So, denaturation first, separation first, then putting on the primers, then extension, that is there. Okay, now one very important thing is that DNA polymerase which is used over here, what do you call it? Does it have any other name? Yes, it has another name, and that is known as TAC polymerase. What polymerase? TAC polymerase. What does TAC stand for? TAC stands for Thermus aquaticus. Thermus aquaticus, which is a bacteria. So, this bacteria has a DNA polymerase which can survive at higher temperature. Do you know that this whole process is happening at a very high temperature? This is denaturation happens at almost 92 to 95 degrees Celsius, okay, which is way too much. Okay, here 55 degrees Celsius, that is annealing, and here 70 to 72 degrees Celsius, which is again way too much. You get it? So, to survive at this particular high temperature, you need to take the DNA polymerase from some other bacteria, that is Thermus aquaticus. That's how you get the name TAQ. Okay, TAC polymerase. So, that is written over here. So, the components, the polymerase chain reaction is a method to amplify DNA in vitro. It is also the method that is there even if small pathogen, if we have a disease, we want to know it. Even if the amount of pathogen is less, we still will be able to identify it with the help of PCR. The components that we require is template DNA. Okay, 2 primers, DNA polymerase, that is thermostable enzyme, and deoxynucleotides, also the nucleotides. Process, you have denaturation, high temperature 95 degrees Celsius, separates the DNA strand. Annealing, where primers bind to the complementary sequences 50 to 65 degrees Celsius, it's actually 55 degrees Celsius. Extension, DNA polymerase, TAC polymerase from Thermus aquaticus, where is it? Synthesizes new DNA strand, that is at 72 degrees Celsius. Repeated cycles will be there, there will be billions of them being formed, and then it is further being used. Okay, with the help of PCR, I'll tell you one more point. With the help of PCR, you are able to do this. Okay, you are even able to amplify the little pathogens which are there, little amount of pathogenic DNA, which is genetic material which is there. Apart from that, you can find mutations also. You will be able to find the mutations also. Please hear to me, this is a very important question. You will see another chapter. You will be able to find mutations also which is there. Any changes in the genetic material also, you will be able to find with the help of PCR. Chalo. Because you put the primers and all, it will generally make a strand of it, but if it is not making a strand of it, probably it has the mutated DNA over here. So, yeah, that is done. Now, give reason for the following. Just before that, let me see how many questions are left. Okay, fine. Give reasons for the following. Alien DNA is linked with the ORI site of the vector in the gene cloning. So, whatever DNA, alien DNA is there, that is from foreign DNA, is been put in the near the ORI site of the vector. Why? I told you all the plasmid needs an ORI site. Now, this question is also asked, what are the features of a vector? So, it should have a ORI site, it should have a selectable marker and things like that, and cloning sites. And what? Cloning sites, with the help of which it will clone. So, over here, ORI site is there. Why do we need a ORI site in the vector, my dear children? Because ORI site is the place where the DNA polymerase will come and attach, and then only what do you call the polymerization would be happening, the replication would be happening. Otherwise, it won't. Now, restriction enzymes are known as molecular scissors because they cut the DNA and the genetic material. Get it? That's why we have what? Restriction enzymes as molecular scissors. Now, DNA ligase is used in genetic engineering. Why? To basically combine the ends of the DNA which have been cut with the help of restriction enzyme. The gel electrophoresis is considered as a very important technique in recombinant DNA technology because you want to separate it, otherwise you will get big DNA. What do you need it? You do not need it, right? I will show you the answers, please don't worry. DNA fragments move towards the anode under electric field through a medium in gel electrophoresis. Why? Because DNA is negatively charged, it moves towards the positive end. Opposite attract and the similar ones repel. So alien DNA is linked with ORI site because it is the site where DNA replication starts. Linking alien DNA to ORI ensures that the inserted gene replicates along with the vector inside the host cell. Okay. Restriction enzymes are called molecular scissors because they cut DNA at specific recognition sites just like scissors, allowing precise cutting of DNA for genetic engineering purposes. DNA ligase is used in genetic engineering. Why? DNA ligase joins the cut DNA fragments by forming phosphodiester bond. Yeah, that point you have to write. They form what? Phosphodiester bond, helping in the insertion of foreign DNA into the vector. Now, gel electrophoresis is considered a very important technique in recombinant DNA technology. Why? Because it is there for separation and purification of DNA based on size. It is crucial for identifying and isolating the desired DNA fragment. Now, DNA fragments move towards the anode under electric field. Why? Because DNA is negatively charged due to its phosphate backbone. The phosphate thing which is there, it gives the negativity. So, it moves through positively charged anode when an electric field is applied in Agarose gel electrophoresis. Okay? Going ahead. Define plasmid. Give an example. I told you plasmid is a small, circular, extra chromosomal. I'll write that. Please use that term. Extra chromosomal. Double-stranded DNA molecule that is distinct from the chromosomal DNA. Chromosomal DNA is in the one that is going to code for proteins and all in the organism. It can replicate independently. It is commonly used as a vector in genetic engineering. Okay, example PBR322 is a widely used plasmid in recombinant DNA technology. Now, insertion of rDNA into the host is an important step. That is, recombinant DNA into the host is an important step in genetic engineering. An ampicillin-resistant gene transferred to E. coli acts as a selectable marker. Explain. How do you explain it? I'll talk about that. Let's keep it for the time being there only. Let's move on to the next question and then I'll come back there. Okay, this is the last question, I think. Yes, this is the last question. So, listen to me. Importance of the following in genetic engineering. Agrobacterian tumor fissions. Until now we didn't talk about that, but it is very important. So I told you about vectorless methods, there is electroporation and there is biolistics and that is gene gun method and there is microinjection. Now if you talk about vectors, the vectors that would be basically passing on whatever is needed to the organism, you have the most important vector, that is Agrobacterian tumor fissions, which is a vector for plants. Which is what? A vector for plants, my dear children. Basically what happens, Agrobacterian tumor fissions is a bacteria which has, okay, Agrobacterian tumor fissions is a bacteria which has its own plasmid, and this plasmid is known as TI plasmid. What do you call it? TI plasmid. What does TI stand for? Tumor inducing plasmid. Tumor inducing plasmid. Now, why is it known as tumor inducing plasmid? Because in this a segment of DNA is there which is known as tDNA, transfer DNA. Whenever this particular bacteria affects the plant, it transfers the plasmid, and the plasmid, the tDNA, causes what? Tumor in the case of plants. So, what did our genetic engineers do? They removed this tDNA. Instead of that, they put our gene of interest. Now, still it will go and infect, but when it goes and infects, is it transferring the tDNA? No, it is transferring our gene of interest. So, it acts like a vector because it is transferring things. You get it? So, it is a vector for plants. Thermus aquaticus, you know, it is used in PCR. I just told you, polymerase chain reaction. Okay, from there only you get the DNA polymerase, which is highly thermostable. EcoHAR1 is a restriction enzyme. What type of enzyme? Endonuclease, my dear children. It cuts within the DNA. It breaks down the sugar phosphate backbone, and the site is 5-GAA-TTC, 3-. Okay, DNA ligase is basically used to ligate the broken DNA fragments which are there. Disarmed retrovirus, again something really important. Retroviruses, they are the vector for human being. So vectorless method was microinjection, not human, basically animals. Sorry. Basically animals. So for animals, you cannot use agrobacterium tumefaciens. They won't be able to do its action there. Disarmed retroviruses, my dear children, retroviruses in general, they can be doing it. Right? Disarmed means we are removing those property of the retroviruses that can cause trouble to us. Just like how we have disarmed this guy, similarly we will be disarming the retroviruses, and these retroviruses have an amazing property of causing the infection to us. So we are removing that infectious property, instead we are putting the gene of interest and letting it go and affect us. So in animals, always remember we have retroviruses for that. Works for you? Okay. Now going ahead, we have this question. Okay. That is insertion of rDNA technology on this thesis. So I'll talk about PBR322. Listen, PBR322 has ampicillin resistant site and tetracycline resistant site. Agreed? Over here, what you're going to do is, you are, just a second. Okay. So basically, okay, here I actually do not need to explain all this thing. It's a very simple question, but I'll explain this first, okay. So what have you done is that there was an E. coli, which has its own plasmid, but now what you are doing is that you are putting ampicillin resistant gene into transfer to the E. coli. You are putting a plasmid that has ampicillin resistant gene. It acts as, how does this particular thing act as a selectable marker? That is a question. How does this act as a selectable marker? As in, for example, E. coli didn't have it first. E. coli didn't have all this first. Right? Agreed? Now, basically, when I put the plasmid that is having ampicillin resistant gene, my dear children, that means this is probably a recombinant plasmid that I am putting. Recombinant plasmid that I am having over here. Probably over here I have the gene of interest as well. I have my gene of interest over here. In this particular plasmid, I have my gene of interest and also a selectable marker. Okay, that is the ampicillin resistant gene. Now, when I am putting it inside the E. coli, what will happen? Nothing will happen. Nothing will happen until and unless basically I treat it with ampicillin. Until and unless I treat it with ampicillin. For making you understand this, I need one more E. coli. So there is another E. coli which has a normal plasmid. Same thing, I am going to treat it with ampicillin again. What will happen if I treat both of them with ampicillin? This guy would be able to survive. This is not being able to survive. Why? Because it does not have the gene. But how did we understand it? How did we understand it? Basically, it is not able to survive, and it is able to survive. That means this has my recombinant DNA. Because this particular plasmid is the one that had the selectable marker that is ampicillin resistant, which is why I am able to understand the whole thing. Otherwise, I would not have been able to understand that this has taken up my gene of interest. Okay, or if you want me to give a better example, here also I have put my gene of interest. Here also I have put my gene of interest. Okay, now if I treat it with ampicillin, how will I understand basically which E. coli has my gene of interest? I do not know. Both of them appear to me the same. When I treat it with ampicillin, what will happen? This one will die, but this one survives. But this one survives. That means what? It basically, because of this particular thing, it is able to survive, right? So isn't it acting as a selectable marker? I am able to select my recombinant plasmid because there is a marker over here. Because there is a marker over here, and that is what? Ampicillin resistant gene. I hope you understand that. Okay, they are going to make me understand selectable markers are the ones that help in understanding that which particular host has taken up the recombinant plasmid. Otherwise, I would not be able to understand it. Okay, now going ahead with the similar thing, very important thing. So basically, PBR322 has two sites, ampicillin resistant and tetracycline resistant part, right? Suppose I am putting my gene of interest in tetracycline resistant part. Now, is it tetracycline resistant anymore? No, this plasmid would not be tetracycline resistant anymore. Now, I take the first plate. I need dual plating for it. Okay, I need first plate where I am having the E. coli. Okay, 2 of them which has taken up the plasmid, 1 of them which has not taken up the plasmid. How do I know that? Basically, this is having ampicillin resistant, this is having ampicillin resistant, and here it is tetracycline resistant, but here I have my gene of interest. This white one is the gene of interest, and here it does not have any plasmid at all. So if it is not visible, I will make it in red. This is the tetracycline resistant gene, okay, and this is the ampicillin resistant gene. Here it's my gene of interest. So basically, these are the different type of E. coli I had. These are the different type of E. coli I have. 2 of them has taken up. 2 of them has taken up the plasmid. 1 of them has not even taken up the plasmid. So how do I understand is that basically I treat it with ampicillin first. When I treat with ampicillin, only 2 will be able to survive, 1 would not be able to survive. So I remove this. And what are these 2 known as? These 2 will be known as transformants. These 2 will be known as transformants. They have taken up, they have got transformed. Now I have to find among them which one has the recombinant DNA plasmid. Which one has a recombinant? It is this one. But how will I be able to identify it? The next plating, where what do? I have the same E. coli, my dear children, over here with my gene of interest over here, ampicillin resistance over here, and this one which has the tetracycline resistance as well. Now I am going to plate it in tetracycline medium. I am going to plate it in tetracycline medium. The one that does not survive is the recombinant one. The one that would not be surviving would be the recombinant one, because the recombinant one will be insertionally inactivated, my dear children, because I have put my tetracycline resistant, instead of tetracycline I have my gene of interest, so it is not resistant to tetracycline anymore. So if this is resistant to tetracycline, that means it has not taken up my gene of interest. So this is what? A non-recombinant. This is a non-recombinant, whereas over here, this one which has my gene of interest, it won't be able to survive in tetracycline medium. That means this is what? Recombinant. You get it? So that's why you need dual plating. Over here, BAMH1 site in tetracycline resistant gene is used for inserting what? Foreign DNA. You are inserting the foreign DNA. The recombinant plasmids lose tetracycline resistance but retain ampicillin resistance. Selection process. Plate transformants on ampicillin containing medium. Transfer survivors to what? Tetracycline medium. The recombinants grow in ampicillin but not in tetracycline. Yes, the recombinants are going to grow into ampicillin but not in tetracycline. Similarly, the non-recombinants grow in what? Both the media. I hope you understood the funda. I know this is kind of catchy, but once you read it once more from your NCERT, it becomes all the more clear. Okay, going ahead. Now, with the next thing, that is biotechnology and its application, the second last chapter. Over here, first question, one of the applications of biotechnology is the production of insect-resistant cropland. Justify the statement with reference to Bt cotton. About Bt cotton, I already told you Bacillus thuringiensis is a bacteria. It has a gene that is known as cry gene. It has a gene that is known as cry gene, and cry gene codes for protoxins. Protoxin, which is kind of inactive in the bacteria's body. Now, when an insect eats this particular organism, that is bacteria, this bacteria goes into the mid-gut of the insect. What gut? Mid-gut, very important point. Mid-gut of the insect, and slowly this protoxin which is there, it will convert into what? Active toxin. Why, ma'am? Why does it convert into active toxin? Because in the gut of the insect, my dear children, there is alkaline pH. There is what? Alkaline pH, which is why now it has converted to what? Active toxin. And slowly it is going to cleave down the epithelial cells, the lining of the gut, and then it's going to die. That's how it works. Okay, this is going to create pores. Now, different cry genes are going to release different type of toxins. So it is very specific. So we have cry 1AC and cry 2AB for cotton bollworms. This is a very famous question for MCQs. Okay, how do I remember it? First letter, okay, first letter starts with C. First letter starts with C, okay. Second letter starts with B, cotton bollworms, Okay, now you might ask, ma'am, corn borers are also like the same, CB. So over here, cotton bollworms are bit large, no? So we have two things. For corn borers, we just have one gene, and that is cry1AB. Cry1AB. So it's 2AB over here, but this is 1AB over here, which is for corn borers. For corn borers, the C here and B here, corn borers. There's only one letter. That's all. You can remember like that. Okay. So basically, this is the thing. You can just go through it once. It will be really helpful for you. Going ahead with the next thing. What are genetically modified crops? Give example. What are genetically modified crops, my dear children? Where basically what you're doing is that you are using genetic engineering techniques, and you are going to put the desirable genes into that particular organism, and it will make the plants what probably pest resistant or drought resistant or salinity resistant and things like that. So GM crops are plants whose DNA has been altered using genetic engineering techniques to introduce desirable traits like pest resistance, herbicide tolerance, improved nutritional content, etc. For example, BT cotton. What is BT cotton? So we studied about Bacillus thuringiensis. When we are putting the genes of Bacillus thuringiensis into cotton plant, we will call this as BT cotton, my dear children. So BT cotton, it's engineered with cry genes from Bacillus thuringiensis, provide resistance to what? Bollworms. It will be resistant to bollworms. Similarly, please remember, there is golden rice. There is golden rice where we have inserted the precursors for vitamin A, my dear children. So golden rice, if someone has this, basically they get vitamin A. So this was introduced because lots of people, okay, you know, rice is a staple food. So lots of people were having, let's say, night blindness issue because of deficiency in vitamin A. So for them, this in the golden rice there were some, so in the rice there were some changes being made to make it to golden rice which had enough amount of vitamin A. Now the next thing is, what is biopiracy? Okay, comment on biopiracy on basmati rice. So what do you mean by biopiracy? Biopiracy refers to what? Unauthorized use of biological resources and traditional knowledge by multinational companies or other organizations without what? Proper compensation to the indigenous communities. For example, America, okay, I think, sorry, US company, yeah. So US company basically, what they did was, they patented rice. They patented basmati rice, and basmati rice is actually produced in India. How can they patent it? How can they take that right that I will only be the person, we will be the only person who would be selling this? You cannot do that, because where is it actually grown? In India, my dear children. So it is actually referring to the unauthorized, they are showing biopiracy. It refers to the unauthorized use of biological resources and traditional knowledge, okay, by what? Multinational companies or organization without proper compensation to indigenous community like us. So a US company, that is Rice Tech, was granted a patent for a certain strains of basmati rice. Getting patency, that means you have the right and everything. Okay, that is, you have the right. Now, falsely claiming novelty, that is, they claimed that we made it. Now this patent could have restricted Indian farmers from exporting traditional basmati rice. If they have got patent, they have got all the right, but it could have done all this thing. India successfully challenged and revoked the patent, protecting its traditional knowledge and agricultural heritage. So they revoked it because it was totally wrong, because they were showing biopiracy. Okay, they were showing biopiracy. Now the amendment, what amendment was being put down, which number of it, so you should know, second amendment was being put down against patency, and all this thing you should be knowing. Please remember that. Okay, similarly in the case of biogas plant, no, who were the organization who created this, who took the initiative for this? So please know that as well. Going ahead. Briefly explain the use of transgenic animals. What do you mean by transgenic animals? When we are altering the genes of an organism, those organisms will be now known as transgenic animals. Okay, got it? So what are the uses of it? For understanding normal physiology and development. For example, I have a gene, I do not know its function. I'm going to basically put that gene into an organism, or probably I'm going to remove one gene from an organism and see what effect does it have. So in that way, I understand what is the use of this particular gene. Okay, so transgenic animals can be specifically designed to allow to study how genes are regulated and how they affect the normal function of the body and its development. Second thing, to study diseases. Okay, so many for understanding diseases such as Alzheimer's, such as cancer and all, we have taken animals as a model. For example, the most common animal is what? Mice, because we have a lot in common. You might be feeling that they're small, okay, but it's not like that. They are very, very useful. Okay, so basically these type of things were conducted for studying of the diseases. Basically, we put drugs on them and see how do they would react to it and things like that. For understanding, suppose this is a gene, if I alter, that's how I get a disease. All of this understanding is being done with the help of transgenic animals. So many transgenic animals are designed to increase our understanding of how they contribute to development of disease. These are specially made to serve as models for human diseases, so that investigation of new treatments for disease is made possible. Now the third thing, biological products. For example, we want certain products. What do? We take the gene, we put in them and let them produce it. Okay, it's a very bad thing to do, but yes, that's how it works. So there was a thing that is alpha-1 antitrypsin. Alpha-1 antitrypsin, my dear children. And why do you need alpha-1 antitrypsin? For the treatment of a disease that is known as emphysema. Emphysema is basically a respiratory disorder. Okay, now alpha-1 antitrypsin was made to be produced from the transgenic organism. Okay, now there was a cow that was, its name is Rosie. Now this Rosie cow was made to produce a milk that was rich in alpha-lactalbumin, which is a human protein. It is a very important protein. So just like that, for getting biological products, we can take use of transgenic animals. So they produce useful biological products can be created by the introduction of the portion of DNA or genes which codes for a particular product, such as human protein used to treat emphysema, that is alpha-1 antitrypsin, and phenylketonuria as well. What is it? Okay. Now human protein enriched milk, that is alpha lactalbumin as well, that is also being produced with the help of a cow, that is Rosie. Now for testing vaccine safety, for testing chemical safety, what do you mean by vaccine safety? For example, we made the vaccine, but before putting into animals, that is us human beings, we put in other animals and see how do they react, something that is very close to us. Okay, we see how do they react to it. So depending on it, this is given down. That is known as vaccine testing or things like that. Then there is chemical safety testing. Basically, we make an organism really sensitive, and then we put little amount of chemical into them and see how do they react. So increasing sensitivity also. So in that way, we understand that okay, if an organism is this sensitive, they might be reacting like this. So we should not. If, for example, they are made very less sensitive, they're not very sensitive, okay, and even if after that, when we're giving some drugs, my dear children, they're reacting very badly to it, that means we understand that this chemical cannot be used on human beings. So this is known as toxicity safety testing, my dear children. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. Toxicity testing in such animals will allow us to obtain what results in lesser time. Okay, next question. What is RNA interference? How does RNA interference help to develop resistance in tobacco plant against nematode infection? So you know what? Tobacco plant is infected, okay, very badly infected by a nematode that is known as Meloidigyne incognitia. Okay, and to stop this, the genetic engineers who were there, they came out with RNA interference technique, where basically what do you do is that, you know how are proteins being made? How are proteins being made, my dear children? The DNA which is there, it is transcribed into RNA, which is known as mRNA, my dear children. This mRNA undergo a process of translation to form proteins. Now what if this is single stranded, that is why it is able to undergo translation to form the proteins. One if basically I silence this mRNA, I do not allow translation to happen? How can we do that, ma'am? Basically, if we add one more strand of RNA, and if it's double-stranded RNA, it cannot undergo the process of translation, my dear children, and this is known as silencing of RNA. And this is known as RNA interference. You're interfering between the function of RNA. This is a defense mechanism that is found in eukaryotes, not prokaryotes. A very important question. It's found in eukaryotes naturally. Okay. Now here we are making the plant resistant to the attack of the nematode. How? We'll see. So we are going to take the DNA that is going to code for the proteins that is very bad for us. Okay, that is the plants. Us means plants. As of now, we are plants. Okay, we have this DNA with us. We are basically going to put this DNA into the plant, but that's a tree I made, but still understand, okay, into the plant. For that I need a vector. What is a vector? It is Agrobacterium tumefaciens, the usual vector. This Agrobacterium tumefaciens will be transferring the DNA over here, okay, and with the help of this DNA which is there, the plant which is there, it is going to make a double-stranded RNA. What RNA? Double-stranded RNA, my dear children. Cool. Now, when the worm Melodiga incognitia, that is a nematode, will come and affect the roots, what are the roots passing from here to here? What are the roots basically going to pass to the nematode, my dear children? It will be the double-stranded RNA. Now, inside, if I zoom in, it looks like this. So, inside they had an mRNA. They had an mRNA which was going to get translated to form the proteins, right? But now, what is happening over here is that this double-stranded RNA is inserted. From here, the one strand comes and binds over here to make it look something like this. Now, it is finally silenced. It cannot undergo the process of translation because it is now double-stranded. So, what tool do we need? We need double-stranded RNA which goes inside, breaks the bond over there, comes and binds with this, makes double-stranded RNA over here, and now it cannot undergo translation, and that's how it works. This is known as RNA interference. Okay, over here. So, first thing, identify the nematode gene. That is the first thing that you do. Scientists first identify genes in the nematode that are essential for its survival or ability to infect the plant. Second thing, you use Agrobacterium tumefaciens to insert the gene. Okay, after you're taking the gene, you're putting into Agrobacterium tumefaciens. This Agrobacterium tumefaciens is going to infect the plant. Now, production of double-stranded RNA in the plant. Once the DNA sequence is inserted, the plant cell begin to produce what? Double-stranded RNA, my dear children, corresponding to the nematode's target gene. Then what? RNA interference in nematodes. When nematode feed on the plant roots, they ingest. Okay, when they feed on the plant roots, my dear children, they are going to ingest what? Double-stranded RNA produced by the tobacco plant. This double-stranded RNA triggers RNA interference in the nematode, silencing the target gene. You get it? As a result, the nematode can no longer form the function properly and cannot cause harm to the plant as well. That's how it works. Okay? Going ahead with the next question. With reference to tissue culture, define the following terms. Okay, explants. What do you mean by explants? So, whatever part of the plant we are taking for tissue culture, my dear, we take a part of the plant and from that we put it in hormones, rooting hormones, shooting hormones. It gives rise to shoot, root, and that's how we get the whole plant. So, the part that we are taking is known as explant. Any part of the plant like a leaf, a stem or root taken for in vitro tissue culture to regenerate into a whole plant is known as explant. What do you mean by micropropagation? It is basically a method of producing large number of plants in a short time through tissue culture under sterile conditions. Okay, so it's a sub part of tissue culture. Micropropagation means it is a technique, it's a method of producing what? So many plants in a very short time through tissue culture under sterile condition. Now, what is totipotency? The ability of a single plant to regenerate into a complete genetically identical plant under suitable condition is known as totipotency. It's the ability of the cell. This cell has the power to become whatever it wants. Whole plant, my dear children, not every cell has the potency, you know. So, totipotency is the ability of a single plant cell to generate into complete genetically identical plant under suitable condition. Now, what is somatic hybridization? Somatic hybridization is basically when I remove the cell wall of two cells. I remove the cell wall, what am I left with? The thing that I am left with is the protoplast. It is known as protoplast in the case of plants. I am removing the cell wall, I just have the protoplast, I am making it hybridized together. Now I have got a hybrid variety of something. Example is potato and tomato, you get pomato. Okay, so that is somatic hybridization, fusion of protoplast from different plant species or varieties to create a hybrid without sexual reproduction. You are not involving sexual reproduction over here. Now, what do you mean by soma clones? Genetically identical plants produced through tissue culture. Okay, now you did the whole process, you got different plants. Are they actually different? No, they're same. It's like identical plants because we are getting it from the same explant. Okay, they're all identical. Genetically, they are going to be same. You get it? So, they are known as soma clones. Okay, so these are the things that you should know. Now, biotechnology has its own applications in the ways as it comprises number of techniques. Given below are the few applications created by biotechnology. Name the technique behind the applications. Detection of pathogen using antigen-antibody interaction. What is this? Antigen-antibody interaction. What is the, okay, which test has this principle of antigen-antibody interaction? It is ELISA. What happens over there? For example, a person has HIV/AIDS. We know that these type of antibodies are produced in an HIV/AIDS patient, so we have it already ready over here. Now, I want to test whether this person has HIV. I'm going to take their sample. In their sample, if we find antigens, they will come and bind over here. If they come and bind over here, that means these antigens is very specific to this antibody, even antibodies are specific to antigens. That means that person has HIV/AIDS. But still, we want to confirm, we have another antibodies, my dear children, which will come and bind over here. And these antibodies are linked to an enzyme, okay? And when we put some coloring agent, what will happen? The color changes. There will be a color change that will be observed. That means the enzyme acts on the coloring agent, and that's why the whole thing happens. So only if the binding has taken place, then only this whole thing would be happening. You get it? That means at the end, if we get to know the color changes there, the intensity of the color, we understand the person has HIV or not. So this is the antigen-antibiotic interaction technique, basically ELISA. Okay. Now, identification of mutation in a gene. I told you, for identification of mutation in a gene, what do you have? You have PCR itself, polymerase chain reaction, a very small line given in your NCERT, but very important. Now, detection of pathogen before development of disease symptoms. I told you very little element of pathogen is there. Still, you want to detect. What do you have? Again, you have PCR. Going ahead, control of Malarial infection in tobacco plants. Control of Malarial infection in tobacco plants. What is it? RNA interference. It is RNA interference. Okay. Now, production of thousands of plants in short duration using tissue culture. This is known as micropropagation. This is known as micropropagation. Yes. Yes. Okay. Everything is correct. Fine. Now, the last chapter, that is organisms and its population, and it feels so hot. Okay, are we ready? Do we need a break? So, I am not taking a break, I am going ahead, okay. If you want to take a break, you can pause the video for some time and come back. But the break should not be very long, okay. So, let's get started. Let's finish off, yeah. Let's finish off biology. So, organism and population, going ahead with what we have is, mention 2 examples for defense of prey against predators in plants. What are the 2 mechanisms for defense? So, how many questions do we have over here? Few more. We can do it, children, we can do it. So, defense of prey against predators in the case of plants. So, plants, you know, like acacia, like cactus, they have thorns. So, they are morphological defense mechanism. Morphology as in they are having structural changes in them, okay, such as thorns in acacia and cactus are one line of defense. Second thing, there are certain plants which basically store some chemicals in them. So, some animals which go and eat them, they get, I don't like it. So, basically it makes the herbivore sick and they won't come again. Again, that is a defense mechanism. Example is also there, Calotropis. Calotropis, which is there, it produces cardiac glycosides which are poisonous. Okay, they produce what? Cardiac glycosides which are poisonous, and it is against what? Herbivores. It is against all the organisms that eat the plants. Okay. Okay. So, these are the things. So, for plants, herbivores are the predators. Nearly 25% of all insects are known to be phytophagous, as in they feed on the plant sap. So, for killing all of them, we have all this thing. Going ahead. The same thing can be asked for animals also. For animals, you have monarch butterfly. Monarch butterfly is highly distasteful because it has some, in the larval stage, it feeds on some plant which has toxic traits. So, which is why it is highly distasteful. Apart from that, some animals, they show camouflaging. Okay, they show camouflaging. That means you won't be able to identify where they are located because of the color they change. So, that is also a mechanism, defense mechanism. Now, write a note on resource partitioning with a suitable example. What do you mean by resource partitioning? For example, me and my brother are staying here. Okay. Now, how do I frame this? Okay, me and my brother are staying. We have Maggie. My dear children. Now, this Maggie, I and him, both of them like the Maggie. So, what my mother or my father do is that they divide the Maggie equally and give to them. That means me and my brother. So, we are able to coexist. Our resources are same. We want the same resources, that is Maggie. But we are able to coexist. Why? Because there is a partitioning being done. That is known as resource partitioning. It is a mechanism that enables closely related species to coexist in the same habitat by using resources differently. It is actually using resources differently, but I didn't have an example like that. So, yes. Okay, so I am given two things. There are resources are being used differently. Okay. Now over here, concept is by MacArthur, my dear children. He observed that 5 species of warbler. Okay, please remember the example, very important. 5 species of warbler were living on the same tree, and they utilized different foraging zones to reduce competition. That means few of them said that we will only eat what is in the lower region. Others said that we will only eat what is there in the upper region. So they have different zones, my dear children, which let them survive on the same plant. Okay, and obviously what is the use of this? This prevents competitive exclusion. Competitive exclusion as in we fight. One of them is excluded, me or my brother dies, that's all. Okay, that is known as competitive exclusion. You are making other organisms extinct. So are we doing that over here? No, we are preventing that over here. When species share the same resource but use it different ways, direct competition is minimized, my dear children. Example, the warblers divided their feeding areas within the tree using different heights and sections for food collection. Significance, it helps in coexistence of species and maintains what? Biodiversity. Okay, now next thing. If N is a population density at time t, B is natality, D is mortality, I is immigration, E is emigration, give the equation to determine density at time t plus 1. So listen, first thing, what do you mean by mortality? Mortality means death rate. Natality means number of birth in a particular time. Immigration means number of organisms coming inside, emigration means number of organisms going outside. So if N is the population density at time t, what is the population density at time t plus 1? There is a change in the time. So how do you represent it? How do you represent it? So there are two equations, okay. Sorry, this is one equation, that is this, that is NT plus 1 is equals to NT, that is the actual population size that we had initially, the density that we had, plus B plus 1, because both birth rate and immigration is going to increase the population size, minus what? Death rate and emigration. That means natality minus emigration. That is the whole equation for NT plus 1. Please know this equation. NT, that is the population density at time t, okay, plus B plus I, that is birth rate and immigration, minus, again bracket, D plus E, that is emigration. That is how you get after time t what is the population density. Over here you have to mention like this only. Now the next question is, which of the above determinants contribute to an increase in population density? What do you think will lead to increase? It is obviously what? The natality as well as immigration. The natality and immigration will lead to increase in population density. So which of the above determinants contribute to decrease in population density, my dear children? It will be what? Decrease as in it will be mortality, and emigration will lead to decrease in things. Okay, going ahead. If a plot of n in relation to time t results in a sigmoid curve and dn by dt is equals to r and k minus n is equals to k, name the type of population growth. What does k represent in the equation? On what condition does the curve become stationary? Okay, so listen, the curve is something like this. There is the population density mentioned over here, that is represented by n. This is t, that is time. Okay, now we are assuming that slowly the population is growing, but since the resources are very limiting, it gets into stationary phase. So there is a carrying capacity for the ecosystem that we are staying in. See, that is represented, carrying capacity, and that is represented by the term K. That is represented by the term K. Okay, now over here the plot is like this. That means you are including K, my dear children, as I mentioned over here. We are including K. That means the population density after time t is equals to the initial density that we have, the reproductive capacity which is there, that is intrinsic rate of natural increase, okay, and then K minus n divided by K. That means the amount of actual organism that can stay in this particular carrying capacity. So what type of population growth is this? This is a logistic growth. This is what? Logistic growth. You know, there is one more growth, that is known as exponential growth, where the organisms are going to grow like this, thinking that there is no resources that are limited. They don't think that the resources are limiting. So when resources are not limiting, my dear children, they will be growing like that. For example, if we talk about bacterial growth in a culture, I am providing them all the resources, my dear children, they are going to keep growing, so they are going to show exponential growth. But over here, since there is a limiting factor, and they have mentioned that over here by showing that there is K, that means it is what? Logistic growth. So name the type of population growth, it is logistic growth. What does K represent in the equation? K means carrying capacity. Now, on what condition does the curve become stationary? On what condition? That is, the community, the growth of the population has reached a level where there is no much resources available, which is why it is stationary. It has reached the carrying capacity. So, the given equation represents logistic growth, which results in sigmoid or S-shaped curve. This S curve is what? S-shaped. Now, meaning of K in equation. K represents the carrying capacity, that is the maximum population size that the environment can support. That is K. Now, condition for stationary phase. The population growth slows down and becomes stationary when N is equals to K, or basically, okay, when N is equals to K, my dear children. When the population growth has reached the carrying capacity, that's when it stops. This happens when resources becomes limited, leading to zero population growth, that is dN by dt is equals to 0. Okay, going ahead. Explain how Mediterranean orchid Ophrys employs sexual deceit to ensure pollination. So, there is an orchid, that is Mediterranean orchid, Ophrys you call it. So, it looks like the female version of the bee. So, what it does is that it appears like that. So, the bee comes to pseudo-copulate with this particular flower, which is orchid flower, which is there. And in that way, it ensures that pollination is definitely happening. Over here. So, Mediterranean orchid Ophrys employs sexual deceit. This technique is known as sexual deceit. You are deceiving them. Okay, and the flower mimics the female bee in shape, color and scent. Male bees mistake the flower for a female and attempt to copulate, that is pseudo-copulation. During this process, pollen grains get attached to the male bee's body. Okay, when the bee visits another Ophrys flower, pollination occurs as the pollen is transferred. The strategy helps in effective pollination without providing what? Nectar as a reward. There is no nectar which is provided as a reward. So, this is not given in NCERT, so we will not follow this particular line, my dear children. Just think of it as an example for sexual deceit. Going ahead, explain the Pearl logistic growth with a diagram and write its mathematical expression. I already showed you the diagram, which is like this. There is a population density which is represented by N. This is T, my dear children, and over here we have a lag phase which slow, where there is a slow growth, then there is a exponential phase where there is natural growth, and then there is a stationary phase, my dear children, where it has finally reached the carrying capacity. This is represented by K. So, this is lag, this is exponential, and this is stationary phase. So, Pearl's logistic growth describes population growth under what? Limited resources. Growth slows as population nears carrying capacity K, forming an S-shaped curve, which looks like this. The equation for this is this. Okay, P is population size, this is growth, this is the carrying capacity, and phase is lag phase, exponential, stationary. This is more realistic than the exponential, obviously, because do we have unlimited resources, my dear children? No. Now, going ahead, parasitic mode of life ensures free lodging and free meals. Justify the statement by listing the special adaptations developed by the parasites. So, there is no use of unnecessary sense organs, so they lose all of it. The presence of adhesive organs are there so that they can stick and basically just survive there. Okay, there is loss of digestive system, again no need. High reproductive capacity is there because we want to increase the number, right? We are a parasite. Now, complex life cycle involving one or two intermediate host vectors to facilitate parasitization in its primary host. So, it ensures that I can be really safe, which is why it has multiple hosts, my dear children. Okay, so these are the things. Going ahead, name the type of interaction seen in each of the following. Clownfish living among the tentacles of the sea anemone. So, you have so many examples, please go read it, very, very important. So, a sea anemone is an organism in which, so they have tentacles, in the tentacles the small little Nemo stays. So, this is the example of commensalism. Commensalism is basically when one organism is benefited, other organism is neither harmed nor benefited. Okay, so over here it is assumed that the clownfish is getting the benefit of protection because these tentacles would be like scaring off some other organisms. Okay, so anemone is not getting much benefit, but the clownfish is, which is why it is commensalism. Now, quail and cuckoo bird during breeding season. So, this is example of brood parasitism. This is example of brood parasitism. Brood parasitism means basically what is happening over here is that the quail which is there, it goes and lays egg in the cuckoo's wait. There is a mistake. Yes, now it's fine. Yes. So basically, the coil which is there, it goes and lays its egg in the crow's nest, okay, and it has evolved in a way that it looks just like the crow's egg, and the crow papa person, it keeps on nourishing the egg. So it is kind of a parasitism known as brood parasitism, my dear children. Now barnacles growing on the back of the whale, again this is an example of commensalism, because the whale is not getting any direct benefit, but the barnacle are able to move around because it is just stuck on the whale's back. Now marine fish and copypods are going to affect the marine fish. It is known as parasitism. It is parasitism, my dear children. Okay, copypods are parasites on marine fishes. Now sexual deceit in Mediterranean orchid, that is Ophrys, even though it is put under mutualism, but they have put a statement that is not all orchids provide rewards. So I do not know whether it is considered to be mutualism or not. I would be telling the answer later on, even though I have put it as mutualism, under mutualism, because it is there like that under in your NCERT flow, but I still want to just go through it once more. This is your crow. Okay, I will be confirming this. I will be confirming this. Okay. Now refer to the graphs given below regarding the growth patterns in organisms and answer the following question. Okay, so curve A represents which type of growth? This is what? Exponential growth. It does not stop anywhere. This is exponential. Curve B represents which type of growth? This is logistic growth. Now which growth model is considered more realistic than in the nature? Obviously this one, because you are talking about the carrying capacity over here. Now when bacteria are cultured under controlled optimum environmental condition, okay, which curve is obtained? Under controlled optimum environmental conditions, my dear children. It is controlled, so it will be again this one, not this one. If you are giving them all the resources and everything, that will be something else. Again, let's see. Now the interactions such as predation, competition and parasitism can be represented by which curve? It will be this one, logistic curve, because the resources are limited, which is why they are competing, they are predating and things like that. Okay, so it will be this curve, but still exponential. Okay, sorry, there's a problem. So it said optimum as in they have enough of things, so it will be this curve. Okay, I'm so sorry, it will be logistic curve. Cool. So curve A represents exponential growth, curve B represents logistic growth, and which growth model is considered? It is logistic growth model. When bacteria cultured under control optimum environment, which curve is obtained? So control optimum conditions means this with unlimited resources, so it will be exponential curve only. Now interactions such as predation, competition, parasitism comes under what? It is coming under, it is these interactions limit the population growth, so it is represented by logistic growth curve. Okay. Now, fill out the tabular column with the appropriate terms describing biological interactions among the species. So, phytophagous insects, and it is going to feed on what? And this is known as predation. Well, it is going to feed on what you know as? It is plants. They are going to feed on plants. Now, Balanus and Thalamus. Balanus and Thalamus, do you know that? They were showing competition, my dear children. They were showing competition. They were both in the intertidal region. Slowly what happened, the Balanus, which was dominant one, they are both barnacles, okay. The dominant one basically made competitive exclusion, okay, made them to do competitive exclusion. That is, they were moved upwards. They moved what? Upwards, my dear children. Okay, you get it? So this is competition that is happening. Now Gauss's law, please go through it once. Now marine fish and copypods, this is parasitism. Parasitism. Now orchid and dash, not the orchid Ophrys, there is another orchid, my dear children, which is there on the mango tree. So on the mango tree, they just grow as epiphytes. They do not take anything from the mango plant. They just want the enough amount of sunlight, which is why they are upwards. So this is commensalism. It is not harming the plant, and it's not even helping it. So it will be this. Fig and dash is mutualism. Fig and wasp, my dear children, wasp is a pollinating agent, and fig is providing a safe space for them to lay the eggs. So it is mutualism. Both of them are helping each other. Okay. So phytophagous insects and plants, it is predation, my dear children. Balanus, Thalamus, competition. These barnacle species compete for space on intertidal rocks. Marine fishes and copypods, they show parasitism. And orchid growing on a mango tree, commensalism. The orchid get benefit, support without harming the plant. Fig and fig wasp, basically they show mutualism. So these are the things that you should definitely know. Gauss's competitive exclusion principle, please go through it. Please go through this also once more, very important example is there. Even cattle igrate and the cattle, what did they show? So all the different examples of each type of interactions, you should be knowing it. I have already done it. You can watch the videos as well. Works. I think we are done with the chapter. Are we? No. There's one more question. So I included it. Yes. So predators are prudent and conduits in nature. What do you mean by that? Prudent as in they think, they know what they have to do. They do not randomly go and eat all the organisms which are in lower trophic level. Because if they do, they would not have food later on, my dear children. Which is why I'll call them prudent. So they are prudent because they avoid over exploitation of prey, my dear children. If they over exploit, what will happen? This will lead to decline in their food itself. You get it? And they always target the weak ones. So in the way when they target weak ones, the healthier ones are still left. You know, the disease won't be spread easily. Now predators are conduits in nature, as in they are channels in the nature that helps in the transfer of what? The energy and everything. So predator acts as conduits in the energy flow of an ecosystem, transferring energy from prey species to themselves and other trophic levels as well. Works. Explain commensalism. Oh, I have explained over here. So commensalism is when one species get the benefit, other is neither benefited nor harmed. Orchid growing on an epiphyte, clownfish living among the tentacles, barnacles on the back of the whale, and cattle igrate and the grazing cattle. So when the cattle moves around, the cattle igrate would be feeding on the small insects. Get it? So Gauss's law states that two closely related species competing for the same resources cannot coexist, and competitively inferior one will be eliminated eventually if the resources are limited. This was seen in abundant tortoise and the goats. So goats and tortoise both ate grass, but the tortoise which was there, it was totally eliminated later on because goats, they dominated. Okay? And with that, we complete the whole 5 chapters. My dear children, I'm so sorry if I basically drained you out. I didn't mean to. So even I have very low amount of energy as of now. Okay, I just want to hit the bed. I'm tired. So you also, I know you might be tired, so take some rest, enough amount of rest, and then only go ahead with anything that you have in your life. Prioritize your mental health as well. Okay, and you are going to do really good. Do not worry about anything. I am constantly praying for you. You constantly put your efforts also. Okay, please let me know if you have any doubts, and I told you about that example. I'll just check and let you know. That's all. Take care of yourself. Bye bye, and do not forget to like, share and subscribe, and put down your love in this comment section. Tell us what you need from us. If anything is good, tell us. If anything is bad, definitely tell that as well. So bye bye, take care, lots of love.